For now, I begin with session 1. For session 1, our chairperson is Professor Subodh Kumar Jain and our rapporteur is Dr. Rashmi Srivastava. I would like to request Professor Subodh Kumar Jain to invite speaker for session 1.
Let me inform that perhaps Dr. Divya forgot that this is the time for the formal vote of thanks, and then we will formally start first session. So, good morning and all. On behalf of Department of Zoology, Dr. Hari Singh Gaur Vishwavidyalaya Sagar, and my own behalf, I extend a hearty vote of thanks to the Chief Guest, Honorable Vice Chancellor, Professor Nilima Gupta, for sparing time from her busy schedule to grace the inaugural session of International Webinar on Neurobiology of Behavior, the Challenge and the Promise for Translational Research. She is the source of inspiration to all of us. Thank you very much, Madam. I extend vote of thanks to Professor P.K. Khare, Dean, School of Biological Sciences, for his presence and support to this webinar. I am grateful to Professor Janak Dulariyahi, convener of the webinar, for organizing this webinar, and organizing secretaries Dr. Rajkumar Koiri and Dr. Divya Rawat, who are constantly working very hard to make this event successful. I would like to thank all the resource persons, especially Dr. Jelena, she is here, delegates, fellow colleagues, research scholars, non-teaching staff, and all those who helped us to make this session successful. Also, I thank all the participants for attending the webinar. I wish a very successful international webinar. Thank you very much. May I now request Dr. Divya to please formally start the session.
I invite our first speaker, Professor Jelena, to talk about her lecture. Please welcome, ma'am.
Here I am.
On behalf of Dr. Divya Rawat, I would like to announce that now we are starting our first session. I myself, Professor Subodh Kumar Jain, is chairman, and Dr. Rashmi Srivastava is report here. The first lecture in this session will be by Professor Jelena Albert Einstein College of Medicine, Rose F. Kennedy Center, Newark, USA. She will be speaking on translating animal biobehavioral research to human psychology and psychopathology. I would like to introduce Professor Jelena Radulovic. She is MD, PhD. Currently, she is a professor at Albert Einstein College of Medicine, Rose F. Kennedy Center, New York, USA. Earlier, Dr. Jelena Radulovic was a professor at Northwestern University, Feinberg School of Medicine, with a dual appointment in the psychiatry and behavioral sciences and the pharmacology departments. In 2013, Dr. Radulovic was installed as the Dunbar Professor of Bipolar Disorder. She is also an associate editor for the Journal of Neuroscience and a senior editor for the journal Neurobiology of Learning and Memory. Born and raised in Belgrade, Serbia, Jelena has been a scholar since her youth. Encouraged by her academic parents, Jelena learned from early on to nurture her intellect and curiously about the world. She is fluent in Serbian, German, English, Italian, and French. During her formative years, she personally witnessed the delineating effect of neurological disease and decided to study medicine with a specialization in neuropsychiatry. Professor Jelena received her medical degree from the University of Belgrade in 1988. Dr. Radulovic studied the molecular and cellular mechanism by which memories of stressful events contribute to anxiety and depression-like behavior. Specifically, her interest lies in neuronal receptor, signal transduction, pathway, and gene response mediating formation and extension of aversive memory, delayed enhancement of fear memory in the aftermath of a stressful experience, social modeling of fear, and the relationship between aversive memories and depression. Dr. Radulovic's long-term goal is to elucidate the key brain mechanisms linking cognitive and emotional processes and identify novel targets for the treatment of anxiety and depression. She has more than 100 peer-reviewed research papers with more than 6,000 citations. Major publications in behavioral brain research, cell reports, neurobiology of learning and memory, cell reports medicine, and several others. She has completed many projects and received grants from National Institute of Mental Health. So this is her introduction, and let me inform the House that her talk initially will be for 45 minutes, then there will be 15 minutes discussion, and that will be followed by the talk again. So with this, may I request Dr. Jelena to please start your talk.
Thank you very much for the invitation, for your kind words, and for hosting me again. I almost feel like home in India now. I hope you can all hear me. I'm very happy to see many familiar faces, but I'm especially happy to see my old students here. I saw on the list that Ravina is here, and Shurbi and Reyes and Dave, who hosted me during my beautiful stay last time. And I will start my talk by sharing, which I don't know whether you can see actually. Can you see my presentation?
No, it's not visible, no.
So I am on the, let me see now. Okay, okay, sorry. Can you see it now?
Not yet, Jelena.
Not yet?
Yes, now it comes.
It comes? Okay.
Oh, that's great.
So this is me 2 years ago, and all of this is a result of hard work by Ravina and Professor Janakahi. I am, as I said, I'm very happy to be here. Unfortunately, I'm in a hotel at the moment, and they will get me out of here at 11 o'clock, so I will go through my lecture in due time, and I will start right away. What I would like to convey today is what is the basic organization of the brain, why we need animal models to study psychological processes when we can study them fairly well in humans, what are the problems and the challenges related to the use of animal models, what do we believe is a good animal model of a mental process, and which paradigms are most commonly used to study psychological processes. And I will start by giving an overview of how we think today about psychological processes. So you may all know, because I'm sure that in the Indian culture, as in many others, people have struggled to understand psychological processes for many, many centuries. And I know that in Europe, in cultures which are closer to mine, people have thought that emotions are in the heart, and others in the kidney, and others in the lungs. And the first who actually realized and hypothesized that mental processes occur in the brain were posited by Hippocrates somewhere in the 4th century of the new era. So he said that from the brain only arise our pleasures, joys, laughter, and jests, as well as our sorrows, pains, griefs, and tears. But where do we start when we study the brain? As you all know, the brain is a vast electrical network of strongly interconnected neurons. However, it is much more beautiful. This is an imaging of distension tensor capture of white matter in a brain, and you can see all these axonal pathways that connect different areas to one another. It is just a beautiful, beautiful network of neurons that conduct a huge amount of electrical impulses all the time throughout our life. The brain is also a regional nodal network. What does that mean? It means that at certain time, as we do certain tasks, particular groups of regions will be active at the same time. So we never have the whole brain being active at the same time under normal conditions. Some groups will be active when we process visual stimuli, others when we translate sensation to motion and to movement. Others will be activated when we regulate the tone to counter gravity, and others when we think about and reflect about our past history, about problems, about past experiences. This is the last network here known as the default mode network. So at any time, we have very organized but regionally specialized activities in the brain. The brain is also a cellular network. The brain contains very specialized cells called neurons, as well as a lot of supporting cells which provide and interact with neurons. And neurons are connected to one another through synaptic connections, which are unique to neuronal communication. So what we have in neurons that we don't have in other cells is that electrical signals generated on one side can be transmitted very rapidly from one cell to another through an entire network. Lastly, neurons are only cells like all other cells, which actually doesn't mean that they are simple. On the contrary, it means that they, inside their subcellular and cellular compartments, contain vast number of interconnected molecules. This is an example of proteins. It is estimated that a single neuron has around 40,000 proteins expressed at any time. However, there are also messenger RNAs, microRNAs as exemplified here. We have lipids, carbohydrates, and many complex proteins. So where do we start when we want to analyze behavior? This is an example that even single molecules in the brain, but also in all other tissues, can perform computations. And although this is a jokingly made cartoon, you can see that a single molecule of a polymer laid as a monolayer on a surface can acquire many different configurations depending on the properties of the environment. And jokingly, this has been compared to brain imaging studies. So you can see here, this configuration resembles on somebody watching, this on somebody listening, thinking, and so on. When we study behavior, we have to assume that each and every one of these organizational units of the brain contributes to behavior, and that if we perturb any of these processing units, we are very likely to impact behavior if we pick the right place, right time, and the right molecule. Now, why did we decide to organize this webinar? And why do we believe that it is important to address the questions of behavior now? This is crucially important because there have been recent advancements which are really changing the way how we do neuroscience research. In the past 10 years, there has been advances in the manipulation of brain circuits to an unprecedented precision. We can, for example, make genetic manipulations in the single cells. We can label entire circuits, but very defined circuits. We can manipulate actually cells where two different circuits meet and connect to one another, and so on and so forth. This is a repetition of the diagram below, but we can also do other very complicated things that we could not have imagined in the past. We can label active cells and manipulate these cells specifically, and we can study many processes in time and in real time, watch how they change. So, before I go to behavior, I would like to just come to and discuss with you some definitions so that we are on the same page when we talk about these things. What is a psychological process? What is it that we are trying to model? There are many definitions, many of them not very clear. I selected here some of those that actually I found the most informative. According to Ricardo Tamayo, a psychological process is a series of steps or mechanisms that occur in a regular way to attain changes in behavior, emotion, or thought. So basically, the best maybe to exemplify what is a psychological process are perception, learning, language, thought, attention, memory, motivation, and emotion. What is the role of these processes? Why do they exist, and what is the evolutionary advantage that they confer? They deal all with information, and this is actual information from the environment. And by environment, I mean external as well as internal. As Dr. Gupta mentioned earlier, the brain processes not only visual and acoustic and other information from the environment, but also signals that come from our bodies, our immune system, our cardiovascular system, and so on. The integration of all this exteroceptive and interoceptive input is organized in our brain through all of these processes by which we perceive changes in the environment, we attend to them, we organize them by value, we store them in the memory system, and then we recall them from our memory systems and try to manipulate them and use them to coordinate our actions in an adaptive way. Now, the question is, why do we need animal models in research? And it is important to address this question. I personally believe that very soon we will probably not even need animal models, but we still do. So we still don't have enough information to make simulations and computational models alone. And we still cannot do many things in humans that we can do in animals. So what is it that we can do in animals? First of all, we can avoid the genetic heterogeneity that we see in humans. We can control the environmental factors and the history of animals, so we can have much better control of the conditions, and thereby we can have much stronger rigor of the research. And the third reason is that we can identify genes and mechanisms in vivo using very sophisticated neurobiological tools that we cannot do in humans. Why do we need that? This is the only way that we can move from correlating changes in the brain and move from correlation to causation by directly showing the relationship between different events. Now, how do we know if an animal model is valid? And here as a field, we don't have a real consensus, but I will show you some examples of what we think about valid or invalid animal models. First of all, an animal model is valid to the extent to which it measures what it claims to measure. So what are the types of validity? These three types of validity were actually put forward by Bill McKinney, a primate behavior scientist and a psychiatrist at Northwestern University, where I first worked when I went to the States. He was working on depression in monkeys, and while developing models, he recognized that three types of validity are very important. Face validity reflects the similarity to a human condition. Construct validity, the accuracy with which the test measures what it is intended to measure. And predictive validity, which is how much, based on an animal model, can we make predictions about human conditions. Now, there are other types of validity, which I will not talk about in great detail. I just listed them here for your information. So we can have construct validity, we can have discriminant validity. All these types of validity are actually much more sophisticated and tuned ways by which we really can dissect how much the measurements that we make are applicable, robust, reproducible, and valid. Etiological validity I will highlight because it is very often brought up, especially in models of psychiatric conditions, because they deal with the etiology of phenomena that we measure. Namely, we're trying to establish whether, if we find the causes of behavior in animal models, whether the same causes cause psychological phenomena in humans. One important property of valid animal models is reliability, which is the consistency and stability with which the variables that we measure are observed across different measurements and laboratories. As a field, we have actually moved quite a long way in establishing reliability in the models that we apply to our research. Now, are these all types of validity equally important? Well, some feel that this is not the case. Mark Geyer and Atina Marco, who have been really some of the pioneers in behavioral research, especially in translational psychiatry research, highlight that predictive validity and reliability are the only necessary and sufficient criteria for the evaluation of an animal model. So if we can make accurate predictions based on animal data, and if we can reliably replicate those data, that, according to Geyer and Marco, is the only necessary and sufficient condition for a valid animal model. Now, there are others who simply think that there are no good animal models at all. They only think that they are useful given a set of objectives and boundary conditions. And I personally find this as the most interesting and important comment and observation by Will Glass. Why? Because animal models, as well as the psychological phenomena and behaviors that they measure, are never adaptive and valid in absolute terms. Every behavior which is adaptive in one situation can be maladaptive in another. Every psychological phenomenon which conveys strong advantages in some situations confers strong disadvantages in others. We will discuss that when we talk about individual behaviors in different types of tests. Now, I would like immediately to say that two biggest groups of behaviors that we study in animals are approach and avoidance. By approach, we consider repetitive behavior and repetitive approach in response to a stimulus. By avoidance, we mean running away from a stimulus or cessation of repetitive behavior in response to a stimulus. Let me put this in simpler terms. What do we mean with repetitive behavior? Well, if we present the animals the food, the animals will go and continuously consume the food, especially if they're food deprived. If the same animals are now, who are consuming the food, are exposed to a threatening stimulus, they will immediately stop consuming the food and will try to find a way to avoid. So basically, these are the two groups. I think almost every behavior that we study in animals can fall in the category of approach or avoidance. Now, so far, this sounds easy. However, this is really not that easy to interpret. There are many reasons why animals approach things, environments, or other animals. They're not only approaching because they like something, which we are commonly led to believe. They may first approach because they want to achieve something, which is what we mainly believe is happening, because we humans are very goal-oriented, and we believe that every other organism is goal-oriented. Sometimes animal can approach due to sheer curiosity, which is a desire to attain information. Sometimes they can approach because they are deprived, so they have a need to achieve homeostasis. Sometimes they approach because they want something, they want to attain reward. Sometimes the factors that organize, that determine approach are emotional. Maybe they feel attracted or drawn to something. They want to experience pleasure. Sometimes they want to experience anger and they attack. Anger and attack is actually an approach response, which has nothing to do with liking or with positive motivation. Sometimes they approach things because they remember it as being pleasurable, although it is not anymore. Sometimes they just approach because they're possibly in a good mood, sometimes because they're used to it out of habit, and sometimes maybe just randomly because they don't know what to do. So there is a series of reasons that could cause one simple behavior in an animal, in a human, or in every single organism that has an organized central nervous system. A similar reasoning can be applied to avoidance. Animal can avoid the situation because they want to avoid painful stimulus. Sometimes they want to avoid the prospect of threat or danger, even the danger is not present. Sometimes they avoid situations that cause them to feel bad, although they are not realistically threatening or dangerous by any other objective measurement. Sometimes emotions are strong reasons for avoidance behavior. Sometimes their memories of past negative experience make them avoid. And sometimes, again, they can avoid either because they're not in a good mood or because it's out of habit. Why do we need to distinguish these different causes? These highlighted processes here, motivation, emotion, cognition, mood, habit, these processes are all mediated through completely different circuits. They engage completely different cellular mechanisms, and they engage different molecular mechanisms. So if we study behavior with the goal to have meaningful translational models for humans, we do need to distinguish and to be as confident as possible why we see the behavior that we see. There are occasions when we simply don't see a behavioral response. And again, these are typically situations when we feel, oh, the animals are maybe too scared, or they don't want to do something, but maybe they lack the motivation, maybe they lack of emotion because they have low fear and anxiety levels, maybe they simply cannot learn or remember something, and that's why they behave completely at odds with what we expect. So as you can see, understanding behavior is a real experimental challenge. Now, the two processes that has been investigated more than any others, I would say, at least in the past couple of decades, have been memory and emotions. Memory and emotions have been studied in tight connections with the brain, maybe somewhere from the beginning of the 20th century, when it was clearly shown that memory is one of the key features of the brain and of what we know as conscious life. We also have learned from the earlier works of William James that emotions are tightly linked to memories, and as he expressed very vividly, as if strongly emotional experiences almost leave a scar upon the brain tissue. And James Papez, the famous neuroanatomist who have identified many of the brain circuits, was one of the first to actually separate the behavioral expression of psychological phenomena and their emotional experiences through the cortex. I'm highlighting this because these are some of the earliest observations on memory and emotion that actually have been replicated again and again and again in our research. I would now very briefly touch on emotion because many behaviors that we will talk about have to deal with emotion. And I believe that they're the most complicated psychological phenomena of all. The reason why they're complicated is because they are really multi-component. They have a strong cognitive component. Emotions are about something. Emotions are linked to events and to very specific situations, people, and experiences. They're not like moods. Their moods are diffuse and you feel bad, but you don't know why and how. Emotions are about something, and there is a lot of cognition, a lot of interpretation of the world in the root of emotion. They have a very strong somatic component. We very often experience strong, especially in intense emotions, they are accompanied by very strong sympathetic activation, sweating, respiration, perspiration, increased heart rate, blood pressure, and so on and so forth. They have a motivational component, namely they prompt the individuals to take action to either approach or avoid or escape or do something to deal with that emotional state. So as we go along in different animal models, we will be discussing to what extent these different components contribute to this holistic phenomenon of emotion, which actually integrates all this phenomena. Now, when we measure memory in animals, we do that slightly differently than we do in humans. But before going there, I just wanted to give you some overview of measurement of memory, which is actually pretty common to both animals and humans. And we can study memory through memory systems, which I will show in a moment. We have different memory systems. We can study through memory types. For example, we have working memory, which is the capacity to hold information at the same time in the brain. But we can also study long-term memory, which is memories of events that have happened very long ago. We can study different memory phases from acquisition, formation, consolidation, long-term consolidation, systems consolidation, and so on. We can study different mechanisms of memory, such as encoding, retrieval, forgetting, and we can actually study memories which are easily or not so easily accessible to retrieval. These dissociated memories are particularly interesting, and I will discuss them in a bit more detail later on. So when we talk about memory systems, in humans, this is a human categorization in declarative and non-declarative memories. Declarative memories are those which are consciously elaborated in the brain. They depend on the cortex, and these are memories of our own experiences or memories of facts. In animals, actually, we can study episodic-like memories, but we cannot study semantic memories because simply we have no way of testing their verbal knowledge. So semantic memories are only investigated in humans. However, non-declarative memories we can test completely in animals as well as in humans. They consist of skill learning, priming, conditioning, so we can do all this, we can model all these processes in animals with very high reliability and reproducibility, and this is very valuable. We can study also in animals as well as in humans sensory memories, attention, perception, long-term memory, and working memory, as well as responses. Now, because we can't talk to the animals, we can't have insight in their subjective experience, we mainly make inferences about their different types of memories through their behavioral responses. We can also study memories by manipulating their different circuits. We know, for example, that early during memory formation, this is in the episodic memory system, which is the really memories of our personal experiences. This is our autobiographical memories. This is what actually we identify the most of all memory systems with. We know that in the early phases of memory formation, the hippocampus plays a critical role. And then as the memories mature with the passage of time, memories become much more cortical. This we see both in animals and in humans. So now we have a pretty good model for analyzing how the cortex in animals organizes memory, and then we can make inferences and testable hypotheses for human research. So whatever we find in animals, please bear in mind, ultimately, the models that we make will need to pass the test in humans. However, it is still very important because we can actually get to those models much easier than in humans. The early times of behavioral research called behaviorism, of which I'm not a very big fan of, but we'll get to this point in a moment, has started through the work of Pavlov first, the Russian physiologist who has established classical conditioning in dogs. I'm sure that you all know about the Pavlov dog who would get food as soon as he would hear a bell, which would cause the dog to salivate in response to the bell, even when the food was no longer there. So basically a neutral stimulus or a conditioned stimulus through associative learning, through predictive learning, that if they hear the tone, they'll get the food, suddenly gets the property of the food, which is the unconditioned stimulus. So this is a very classic conditioning experiment to study conditioning processes in animals. Now, what is important in conditioning is that the animals associate they make predictions about the relationship between different stimuli. There is another type of learning in which animals learn to make predictions about how their own behavior controls the environment. And this type of conditioning is known as operant or instrumental, and it was especially grounded in the work of Edward Thorndike. So we use still a lot the models of Thorndike's work through by studying avoidance. So the animals have to perform a task to avoid the stimulus or have to approach a task to get a reward. In classical conditioning, they don't need to do anything. So the body does everything on its own. In the context of this work, it's worth mentioning Skinner, who was the father of behaviorism, and who thought that human and animal behavior can be entirely explained in terms of conditioning without any appeal to thoughts or feelings, and that psychological disorders are best treated by altering behavior patterns. This view has been prominent in the field for decades, and fortunately has proved to be wrong, because as you all know, even intuitively, thoughts and feelings are actually important organizing systems for behavior. And honestly, I think I personally study behavioral mainly because I would like to use it to make inferences about higher cognitive processes rather than of my personal interest why an animal has moved from here to there. Now, the danger of that, I have to say, is that very often we assign animals a lot of human features and we tend to anthropomorphize. That's the other danger of now taking the opposite view. So we need to really get a fine balance to make consistent predictions and really reliable interpretation of the findings. So this is a summary. What are the key differences between classical and operant conditioning? In classical conditioning, the dog hears a sound and the sound predicts the arrival of the food and the dog salivates, right? There goes the bell, it's time for food. In operant conditioning, the animals learn that they have to press a lever to get the food. So they have to do something. We, as a field, we have worked a lot on a paradigm called contextual fear conditioning. And this is what we do a lot in my lab, and this is what Professor Jelena has done, and we've worked on that together and have some papers together. And what makes this paradigm, what made it very, very popular in the, probably starting from the 90s of the past century, is that it is very fast. Fear conditioning, known as contextual fear conditioning, occurs if we put the mouse in a box and deliver a shock. Only 3 minutes in this box, followed by a shock, will make the mouse to freeze 24 hours later. This is the behavior of the mouse in the box on the first day. You can see the mouse is all over the box. It is sniffing. It stops occasionally somewhere to explore certain corners of the box, but it is quite active. The next day, after delivery of shock, the mice hardly move. They sit in this one corner of the box and don't move. They show very strong freezing behavior. This paradigm has gained a lot of popularity because of the rapidity of learning. Namely, the paradigm was used a lot to study the molecular basis of behavior and the molecular basis of memory. If we were to use paradigms where learning occurs over multiple trials, we would not know where, when, and when to look. Here we know exactly when the learning process starts. And if we take the mouse brains or look at the activity in the mouse brain over these 24 hours, we know that most learning relevant processes will take place in this time frame. So that has proved to be very, very important, and a lot of what we've learned about learning and memory has been due to the application of either contextual fear conditioning or passive avoidance learning, a similar paradigm that can also be achieved in one trial. We can use passive avoidance and fear conditioning to also study another type of memory, a very interesting type of memory, namely memories that cannot be easily remembered. And these memories are called state-dependent memories. This is an example that I want to show you what we mean by state-dependent memory. If an animal is drug-free and learns that in response to this shock, it will run away to the other compartment, we test the animal and it will indeed right away the following day. However, if we put this animal on a drug, which was not present at the time of training, the animals will not remember this information and they will stay in the shock compartment. On the other hand, if we use a drug during training and then place the animals drug-free, they will not remember, but they can only remember if we put them back on drug. They can only remember if the brain is in the same state at retrieval as it is during encoding. And this is valid to both situations, whether they're on or off drug. These models of memory are very interesting because they are showing us that a lot of information can be formed and retained in the brain, even though we cannot access it at all times. And we are now using this paradigm quite a lot in the field to try to unravel memories that we believe are still hidden in some parts of the brain and subconsciously affect behavior. Now, this is the time when I would normally make a break, but I would like to ask you whether there are any questions at this stage. And you can, if you would like to ask me, you can just ask, unmute and ask me, or you can send me a message through a chat if you prefer. Okay. Now, sorry?
Madam, I have got one question.
Yes, please.
I often wonder whether prokaryotes, unicellular organisms, do they exhibit memory, behavior, and emotions, or whether it is the property only of multicellular organisms?
So I couldn't tell about emotions. I don't think so, because I do believe that a central nervous system is required for the experience of emotion. And not only a central nervous system, I actually think that a cortex is required for the experience of emotion. So, because consciousness, emotions and feelings require us to be aware of them. And without the cortex, if we remove the cortex, we know for sure that all these subjective experiences are gone. We know that through lesions, through injuries, through cancer in the brain. So we have pretty strong evidence. I can never say absolute, but pretty strong evidence that emotions are conscious phenomena requiring cortical function. However, learning and memory is very fundamental to every cell. So prokaryotic cells, amoebas can learn. We can see that if they are exposed to toxins in their environment, they will avoid those parts of their environment for a long time. So in a way, it's like the immune system. You see, it's not a brain. Immune cells can remember, although they don't have brain. Cells have different mechanisms to form memories. They can form memories by changing their cytoskeletal motions, by changing their epigenetics, and so on and so forth. However, for multicellular organisms to have this integrated coping and to have a psychological process and coordination between input and behavioral output, the brain is, the nervous system is essential.
Madam, is it possible that, as we can see, that there exist predator-prey relationships in case of unicellular organisms like amoeba or paramecium? So predator-prey behavior is there. Now, as we have got a very intricate relationship between astrocytes, glia, and neurons in case of multicellular organism, which play a role both in neurobehavioral response as well as immune system. So is it possible that there exists such type of some system, even in case of prokaryotes, irrespective of the fact whether they're unicellular organism? Because the moment you are saying immune function means although they are unicellular organisms, there has to be something to regulate the immune system also, because it's only unicellular organisms.
Right. No. I mean, I think that the memory can be a property of every single cell. But emotion, I don't think so. So this is where I think that we put memories and emotions very much apart.
In your knowledge, any group in the world which is working on such type of research with respect to prokaryotes, behavior, emotions, memory? Any group in the world?
Yeah, many groups in the world work on prokaryotic behavior in terms of memory, but I don't know any group that works on emotions in unicellular organisms, whether prokaryotes or eukaryotes.
So that means that all these phenomena are only exist in multicellular organisms,
Not only in
or eukaryotes
multicellular
only,
invertebrates who have developed cortex.
only vertebrates, not in
Yeah.
invertebrates.
Right,
Okay,
because they do require conscious awareness. Now, that is to say, it is possible that some emotional primitives exist also in organisms who don't have cortex, but not in the way as we experienced as in humans. So there have been several types of work talking about 5 different types of consciousness. And that's really critical to emotion. That's really critical to emotion. So we can have consciousness, for example, an animal can be conscious about its all integrated body. On the other hand, it is not likely that they have a concept of self. I think that the only, at least based on some really strong experimental data, there is some agreement that self-consciousness is earliest detected in primates. And even that has been debated or not, but in a way, that level of emotional experience of the self is assumed to be at the level of primates and humans. However, having said that, it seems likely that animals do experience pain. Now, that's an interesting thing. How do we define that? So how does one experience pain without the notion of self? Because, you know, from a human standpoint, we always feel that pain is something that hurts me. So from a mouse perspective, what does it hurt? It says that a leg hurts, but that's not me. A leg is not, you know, we don't really know. I mean, there have been some actually interesting models with artificial intelligence system trying to figure out how can you model pain without the idea of self. And I don't think we have definite conclusions about that. But we certainly see pain-related behavior in animals. So it is, I mean, from a human perspective, it is very, very difficult to imagine any kind of subjective experience without the notion of self. So that's, but I don't think I can contribute more to the debate.
Dr. Rajkumar, I have a question with ma'am. Am I audible?
Yes,
yes, ma'am,
please.
you are audible.
Yeah, Jelena ma'am, I have a question. Like, we think that the humans are the highly evolved and the brain is more developed. But somewhere I read that octopus is a species has a very well developed central nervous system, and then they help in their camouflage function also. Is it so?
Yeah, so, you know, it's hard to say what do we mean by developed, because in terms of numbers, there are many actually species that have more neurons than the human. Elephants, dolphins, they have many more neurons than we have. However, the thing is also where those neurons are and how they're organized. For example, in elephants and in whales, a lot of their brain power and neuron power is dedicated to organization of movement, because they have these massive bodies, and a lot of neuronal activity is just dedicated to organize and coordinate their motion. In octopus, actually, it's a very similar thing. So octopus has to coordinate in very sophisticated ways their movement, although they actually make a lot of very intelligent decisions. So behavioral experiments in octopus show that they are highly adaptive, and that they use very creative ways to find solutions. However, the density of cells and cortical organization that you have in humans, I don't think that that exists in other animals. which is not to say that that makes us superior in every way. It just means that all these conscious phenomena are much stronger in humans. We are far inferior to many, many other solutions than other species, right? I mean, it is just when it comes to thought and simulation and representation of the environment and these really strong conscious subjective experiences, I do believe the complexity of the cortex is that what endows us with language, with all kinds of other forms of strategies that many other species do not have available.
Madam, here I would like to add one thing. For example, in case of certain invertebrates, if I'm not wrong, like loligo and all, they have got ink glands, and whenever they encounter any predators, in order to escape the predators, they release ink gland in the water system, and they evade the predator response. So the behavior is there. Now, how it is regulated is again, I mean, a subject of research, whether they're having any neural system or not. But they do exhibit such type of behavior. Certain invertebrates
Oh,
are there,
absolutely.
other than octopus and
No,
all. Yeah.
absolutely. I mean, I'm not trying to sound like humans are great and so on. No, not really. It is just so that we are talking about psychological phenomena, and we only have access to psychological phenomena directly from humans, right? For everything else, we have to guess. And if you remember, when in Japan they invented those little dolls, Tamagotchi dolls. I don't know if you know that. That was famous maybe 20 years ago. And people, it's amazing how easily we can assign human properties even to inanimate objects. These dolls would ask for food, and so, they were programmed. And usually they were owned by teenage kids. And these kids were crying if these dolls would die. The dolls would suddenly stop working. And they looked at them as if they were living beings, So it's very easy if we see something resembling human behavior to give human properties. But the thing is, it's very difficult to know. So I will not take a stand here as if we know what exactly is going on in their brain. And they clearly have some very basic motivational memory and drive forces. But when it comes to more complex things like creativity, thinking, emotion, I would not be inclined to give to those labels to invertebrates and prokaryotes. But that's me.
Okay, thank you, ma'am.
Thank you, ma'am.
Okay. Now, can you see again my screen?
Yes, ma'am. Your screen is visible.
Okay, good. Now, when we look at all kinds of processes, so we talked about behavior, and behavior is an exophenotype, right? It's something that we see, something that is overt. There are also a lot of endotypes. So what are endophenotypes? These are these invisible, measurable alterations that we see somewhere at the interface between a genotype and a behavior. And endophenotypes were very popular in research at some point, and they're actually still a very important point of research. What are endophenotypes? They're simple clues to diseases. So that's when they were actually first put forward. The idea was to use endophenotypes to look for diseases, but they can actually use to also look at all other functions and dysfunctions in the body. So what are endophenotypes? Endophenotypes are nowadays actually called as biomarkers, This can be changes in the blood, changes in the skin, changes of hormonal status, of RNA expression, of connectivity, of any type of measurement that we see which is not subject of overt behavior. So typically when we make assessment about brain function and psychological processes as well, we try to look at both behavior as well as changes in the brain, in the body, that could be a reliable correlate of that behavior. So we are trying to find really different types of changes and put them all together and see how this all comes to play. Now, typically, we can study behavior to understand normal psychological processes, but even more attention to animal models has been brought through trying to understand psychiatric disorders. And throughout the decades of research, we have used different models. Pharmacological models are the oldest. For example, we have used drugs to induce abnormalities of behavior that could resemble schizophrenia or autism or hallucinogenic drugs to see what happens to model hallucinations and so on. Very popular in the last century were genetic manipulations. We use them ever since and even more now. However, they're more and more combined with more modern circuit manipulations. In any case, this is now almost accepted as a norm, but I remember when we started working on this, this was a huge revolution in the field. The field went from electrophysiology and recording currents in the brain to very massive genetic research on knocking out different genes, then knocking them back in again, then trying to identify single genes to model disease, or trying to inbreed different animals based on certain behavior and try to get different lines to study such behavior. Then there were models in which we would combine genetic and pharmacological approaches. For example, sometimes gene knockouts wouldn't show any overt behavioral problems, but then if we would put some pharmacological treatments on top of them, then we could see overt phenotypes. And also, we have very often combined genetic and environmental or environmental manipulations alone, and especially prominent were models of acute and chronic stress. So these models are still used a lot. They are becoming more and more sophisticated and implemented more and more variables, such as predictability, unpredictability, and so on and so forth. And typically, we can also see very often, even when genetic manipulations alone do not result in certain behavior, if we stress the mice, suddenly we can see a lot of overt behavioral phenotypes. So the most common models that we have used are animal models for anxiety disorders, depression, autism spectrum disorders, and schizophrenia. And what I will show you soon is I still decided to keep these models because I want to show you how the world and our understanding changes. And many of the models that I will show you, which were thought of as the foundation of how we test psychiatric drugs and how we study these different states, are now actually completely not in use anymore. And I know the National Institutes of Mental Health is not even funding projects that are using these types of models. So there has been a very big change in how we see these behavioral models. Fear and anxiety have been the most studied of all because they're fundamental. They occur in response to really life-threatening stimuli, and even in organisms that may or may not experience fear, a lot of behaviors which is very consistent with fear and anxiety can be observed. I chose these paintings here for two reasons. You can see the different approach to fear. This is Edvard Munch, who focuses on the behavioral expression on the screen. Paul Klee is more consistent with the model I showed you, where there is, I would call this is the expression, behavioral component, but they're all the other bits and pieces of motivation, cognition, and everything else. Now, when we talk about fear, we are actually talking about one of the most important emotion in humans, and we don't really know whether animals experience fear, but they certainly show behavior which is consistent with fear. Now, in the example that I will show you here, this is a mouse whose receptors for odor processing have been genetically modified so that it cannot perceive the smell of a cat. Normally, perceiving the smell of a cat is an innate psychological process in mice, so odor perception. This mouse cannot smell the cat, and these animals actually cannot survive. So they didn't show that in these photos, but they showed that in the results section. Obviously, detecting threat, detecting danger, and developing an emotional response to that, such as fear, prompts very rapid behavioral adaptations, such as running away, hiding, escaping, and so on. So these behaviors are essential for survival, and they're really varying depending on the proximity of the threat. For example, if something which is potentially threatening is far away, animals usually don't rush away. They're looking, they become a bit more cautious, but they don't show dramatic behavior. And as the threat becomes closer and closer, they go from freezing to escape and to fight and flight. So they show a whole range of different behaviors just as a function of the proximity of threat. So if that's normal fear, what do we call pathological fear? And pathological fear is typically a strong emotional state of fear in situations that are not really threatening. Agoraphobia, fear of open spaces, is very common. Claustrophobia, fear of enclosed or constrained spaces. Fear of spiders. More and more prominent are social fears and social phobias, such as fear of public speaking. Okay, this is fear of height. I wouldn't call this not dangerous. I mean, I personally would be terrified, so I wouldn't blame this person if he experiences fear. But as you can see in all the other examples, there is no real threat, and yet the emotional experience of fear can be devastating. So excessive fear can cause, has been in the root of many anxiety disorders, and their core symptoms are excessive, persistent, uncontrollable, irrational dread of everyday situations. This is just illustrated in a poem by Lewis Carroll, which I usually use for my students because many scientists use the word boojum in their titles of papers, so just wanted to tell them where it comes from. So what happens in disorders? It is not that the fear quality is different or the anxiety quality is different. What changes is the intensity of symptoms, is the duration. The source is absolutely irrelevant, and there is compulsory action which is prompted to relieve symptoms. What we mean by compulsory action, the animals may start grooming suddenly, or rearing, or just do some really not very meaningful actions just to relieve the tension. Now, in the States, I don't think in Europe, but in the States, psychiatrists are going back and forth about trying to categorize these disorders. According to the latest Diagnostic and Statistic Manual, which is the American system of organizing psychiatry disorders, they are separated into anxiety disorders, obsessive compulsive disorders, stress-related disorders, and dissociative disorders. All of these disorders are accompanied by very severe anxiety symptoms. The reason why they're separated is because they involved differently motivational components, such as in OCD, or memory systems, such as post-traumatic stress disorders, or dissociative symptoms, as seen in dissociative amnesia. Again, a lot of disorders have anxiety or excessive fear as a core symptom. How do we test that in animals? These are the tests that we have used over the years, and they have been really scrutinized more recently. And I could say probably rightfully so. So most tests that we have used are based on the animal's innate preference for closed relative to open environments. So if the normally animals stay enclosed, but occasionally come out in the open. And in this light-dark box, they would be mainly in the dark, but occasionally they go to the light and so on. If animals are terribly anxious, they will never go in the light or in the open. They'll just stay at the closed, and they're not even terribly active. There are also tests based on a startle response, and these tests are actually have proved to be more reliable and more translational for humans, because human startle has been actually found to be quite reliable and robust as well. Startle is simply a rapid reflex response to a sudden noise. There are also tests based on burrowing behavior. They are not really translatable to humans in the sense because humans don't usually go and bury a threat that can easily be avoided. On the other hand, they can model pretty well some OCD-like symptoms. One really big problem with this type of behaviors, though, is that you can test them once, and that's it. If you repeatedly put the mice in these boxes, their behavior will completely change, so they don't show any consistent preference for one over the other. They'll become habituated, they'll learn that there is no danger, they'll start going randomly in all of the different compartments. So that's really a very big disadvantage. They they can't measure certain states over time. Now, there are other ways for us to induce fear and anxiety. Stress has been one of the most common. Others are based on genetics, other on pharmacological injections of different drugs. But the models which have proven to be the most robust and reproducible are learning models, especially models of classical and operant conditioning. So once we implement learning and memory, then we get to models that we can test actually over a much longer period of time. And this is a big advantage. And as we mentioned, emotions are about something. So by introduce learning, then at least we know the sources. And we can test, for example, whether fear is induced to relevant stimuli or to irrelevant stimuli. We can look at the duration, we can see on the persistence, and we can test many, many other things. But again, as I said, most of the work has been done in models in the past, so this will probably become soon very historical, but even that is valuable because if you go in this field, much of the literature that you will find will be using this type of apparatuses. Now, this is an illustration where tests like this have shown to be at least consistent and of some etiological validity. The stress response is the response to sudden and strong environmental change, and it involves release of corticotropin releasing factor to the pituitary, and then release of adrenocorticotropin hormone in the circulation, which acts on the adrenal gland and leads to the secretion of the stress hormone cortisol. Cortisol acts on glucocorticoid receptors to organize behavior and a huge number of other metabolic, immune, and other functions. So what does it do in the brain? If we knock out the glucocorticoid receptor specifically in the brain, so to isolate its brain actions relative to all its other actions, what we see is that these animals, relative to controls, actually spend much more time in the lid compartment. And so they spend much more time and they are very fast to get out into that compartment relative to the control animals. Now, you can see this is not a stressed animal. This looks to be like some innate change of preference. This has been interpreted as reduced baseline anxiety in these animals. Quite frankly, I have to say this is a huge number of animals to see a significant effect. So this hasn't been as robust as it has been thought. But the really unfortunate setback for this study is that experiments were really not done under conditions of stress. So if these animals were stressed, probably the findings would have been much stronger. Nevertheless, there is something which is important, which is that a very similar pattern is seen in a completely different test. This is a circular maze where the animals are either in the open or in the closed parts of the maze, and you can see the controls are all in the very little in the open sections. However, the knockouts are quite a bit in the open sections. And this is another important control, namely that their total number of movements is very similar. So this is important because if the genetic knockout will impair motor behavior, then the animals don't move because they can't move and not because they have a preference or they're in a certain emotional state. So this is really important. But I put here this paper. This was really a huge paper when it came out in 1999 in Nature Genetics. This was viewed as one of the revolutionary genetics and behavior papers at the time. This is a paper from my own experiments, also from the same era, 1999. This is a knockout of the CRF receptor 2 in the brain. We can see that control animals are moving. This is a dark light box. So we put the mice here in this dark chamber, and then they slowly go out, and then they spend time outside. These are the knockouts. As you can see, they hardly get out. And CRF is actually acting up here at the brain level. So we can induce a state which resembles pretty high anxiety using genetic manipulations. Now, what do we do with depression? Depression is very difficult to differentiate in animals from anxiety, although they are quite different entities in humans. So I will really highlight some elements which are supposed to be helpful in differentiating the two. First of all, I would like to say that probably the strongest models of depression currently are those induced by social stress. Interestingly enough, mice are very resistant to physical stress in developing depression-like behavior. You can stress them. I mean, people are using 6 months of some intermittent stress to induce depression in animals. That's really a very long time. But if you expose the mice to social defeat, in about 11 to 14 days, they develop actually a series of new behaviors which they have not exhibited before that. One important behavior is they start to avoid not only the mouse that defeated them, but also they start to avoid social interactions with other animals. That is something seen in various psychiatric conditions, including depression. Now, they also avoid open arms in the maze, but as we noticed before, this is impossible to distinguish from anxiety-like behavior. So here, this test is really not very useful to model any kind of depression. But there is another test which is actually quite relevant, and this is a test based on anhedonia. One symptom of depression is that many patients stop finding or experiencing pleasure in response to stimuli that they used to experience pleasure before. So there's really a change, and this change goes downwards in experiencing pleasure. And this is done in mice by exposing them to things, foods or drinks that they usually like. So mice really like to consume sweet solutions, sucrose solutions. However, after chronic social stress, they will actually start equally drinking sucrose or water. So they do not show the preference as they would without any stress. The two classic tests of depression, floating in a water cylinder or hanging on when suspending on the tail, are now completely out of use in most research in the States because they have their validity has been challenged on at many different levels recently. So you will see still probably around the world and in some labs these tests are still used, but very much less than they used to be before. Tests of social behavior, though, and social avoidance are used significantly more. And tests of behavioral despair, which actually were reasonably good tests and very valid tests for depression in the past, are not used because of ethical reasons. Because to get to learned helplessness is a state in which animals learn that no matter what they do, they cannot avoid shocks. And to get to that stage, they need to undergo many repeated and relatively strong shocks, and this is not allowed anymore. So they are not used at this time. However, if you would have seen an animal in this state, that's probably the closest one could see to depression. In addition to this test, models for autism spectrum disorders have gained a lot of attention recently. First of all, because interest in neurodevelopmental disorders has increased as more and more kids are diagnosed with autism spectrum disorders. And the core symptoms of these disorders are deficit in social interactions and communication, restricted interest, stereotype movement, and anxiety. So you can see here already, we have social interactions and anxiety, which already overlap with the test that we have used in anxiety and depression. So what are we left with when it comes to autism? Communication as a very important differentiating behavior and test. In addition, if lack of social behavior, communication is combined with stereotype movements, anxiety or seizures, then the cluster of these behaviors is used as a more or less valid model for autism spectrum disorders. Cognitive function, that's important, is typically preserved. Intelligence and problem solving are typically preserved. Only in some models we can see cognitive deficit, which could resemble mental retardation. Oh, this is too long. I will skip this. But whoever is interested in learning something about autism spectrum disorder, especially Asperger's syndrome, I recommend the book The Curious Incident of the Dog in the Nighttime by Mark Haddon. Beautifully written and completely to the point of what means to not understand social signals from your environment. Well, no, I'll say one, for example, how they cannot understand metaphors. Like I laughed my socks off, or he was the apple of her eye. So they take each and every word literary, and all these inferred meanings and metaphors make no sense to them, and they find them very, very confusing. For a long time, autism spectrum disorders were modeled using pharmacological tools, such as injection of valproic acid and inflammatory agents during pregnancy of the mother. Inflammation is still used, valproic acid very rarely nowadays. Very popular, there are genetic models, and typically they're designed based on backward translation. So mainly, there are autism spectrum disorders in which different genes are identified as strong candidate genes and as strongly associated genes. And then what we do, we analyze one gene after another and look what their role is in development and behavior. This approach actually has proven really good for understanding some manifestations of autism, but also for understanding the role of many molecules that we didn't even know in the past and how they function in the developing and in the adult brain. Hormonal models such as maternal hypothyroidism are not used anymore because they are not maternal hypothyroidism is not seen as a major cause of autism as it used to be in the past. So how do we study social behavior? Remember, I showed you social stress and social defeat. But now on the bright side, sometimes we do nice things to animals and we test how they like to interact with one another. And normally they do like to interact. Although mice are not seen as very social animals as rats are, they actually engaged very readily in interactions, either if they are just presented with the conspecific, and you can also see it if they have a choice whether to interact with a mouse or an inanimate object, they would always choose a mouse. And interestingly, they very often shift attention from a known to a novel one, which also shows that they can recognize and differentiate familiarity from novelty. So they can recognize the feature of individual animals. And many of these aspects of social interactions are impaired in mice which have genetic deficits, which are common to autism spectrum disorders. Other things that we measure are ultrasonic vocalizations, which rodents use to communicate to one another. Also grooming as type of stereotypic behavior. We see seizures. In many of these neurodevelopmental models, we see epileptic seizures. And finally, when all of that clusters together with anxiety-like behavior, the idea is that the model is reasonably valid for studying autism spectrum disorders. This, however, is an example that this doesn't have to be this straightforward, and that many of our research in animal models has resulted in failure to be replicated in humans. This was an example, the famous social love hormone oxytocin. We know that oxytocin is needed for social interactions, and yes, if we knock out the oxytocin receptors, the animals cannot habituate in their interactions. They do not recognize the new mouse. They have very short duration in their interactions and so on. So there were many clinical trials trying to use oxytocin in autism models, and all of them have failed. So this is an example of translation gone wrong, and how animal models actually do not necessarily need to be translatable to humans. After this finding, I have to say, looking at the social interactions has become really not as frequent in research of autism as it used to be before. And many of the studies are now just looking at gene development changes that don't even look at behavior, because the idea is that many of these social behaviors are species specific and not translatable to humans. I don't think that this has been now taken generally for everything, but for many of these behaviors, actually, the field has abandoned the use of social behavior. There were also models of schizophrenia, and you can imagine that these are really difficult to model. Schizophrenia is a disorder of thought and reality perception, and we have no idea how animals perceive the reality and how we can model their thought disorder, how they develop hallucinations and so on. So, you know that schizophrenia in patients shows positive symptoms, hallucinations, delusions, disordered thought, negative symptoms, which are flattening of affect, poverty of speech, deficit in social interaction, and anhedonia. So as you can see, all these symptoms are very similar to depression, and it's very difficult to differentiate. And they also have a lot of cognitive symptoms, such as impairments of attention, working memory, and executive function. This third cluster of symptoms is most reliably modeled in animals, whereas positive symptoms, we just assume that they develop them if we inject them with hallucinogenic drugs. All we see, however, behaviorally, is that the animals start behaving erratically, they become hyperactive. I just wanted to show you in one, I will skip these little things, but how strong some of these behavioral phenotypes can be. And this is actually a finding that was very counterintuitive, and yet, unlike the oxytocin finding, it has opened a very new avenue in schizophrenia research. Namely, the dopaminergic hypothesis of schizophrenia has dominated the field over a long time, and most drugs, which were developed based on animal models to treat schizophrenia, were based on interference with the dopamine system. However, when the first knockout of the NMDA receptor 1 subunit was made, this animal was found to have many symptoms that resembled schizophrenia, such as anhedonia, lack of social interaction, lack of interest in conspecific. You see, these are wild-type animals. They always cluster together. They nest together, and all these schizophrenia animals, all these knockouts were housed separately. Their mating frequency was very low, as it is seen in schizophrenia patients. And what was interesting is that many of these behaviors were reversed when the animals were put on clozapine, which is an antipsychotic drug. What was important is that this finding showed that actually glutamate plays as important role in key deficit of schizophrenia, and even more than dopamine. And most of the drugs that are nowadays tested are based on some modulation of the NMDA receptor in addition to acting on other subunits. And lastly, I'm trying to see, I don't have a watch. Okay, I'm good. I wanted to mention to you something about state-dependent memories, which I already talked about, which are memories which are best recalled if the brain is in a similar state during encoding and retrieval. Otherwise, we have no evidence that such memories exist. And the reason why I'm bringing up this is because models of state-dependent memories can actually help us understand phenomena which are really difficult to understand even in humans. And these are phenomena of dissociative amnesia. Namely, sometimes extremely stressful events cannot be remembered for a long time and then surface at some point in individuals' lives when it is very difficult to verify whether these memories are even accurate or not. Experimentally, the first example of state-dependent learning and memory was actually reported in Chicago, and it was a mistake. In 1937, Jordan, who was a student in Almar Kallar's lab, was conditioning dog reflexes to tone. So tone, shock, muscle construction, tone, shock, muscle construction. But to make things simpler, he put the dogs under curare. And curare is really a very important, potent psychotropic drug, which is actually really changing, especially the cortical, suppressing cortical processing of information. So what happened is when the dogs woke up and the curare effects were gone, Jordan could not elicit any of the reflexes. So he thought that the animals didn't learn anything. But then by mistake, he used the same dog, put it again under curare, and all the reflexes were there. So obviously, on drug, reflexes were there. Off drug, reflexes were not there. And in this case, it means that when certain parts of the brain were active, we could recall the memory and vice versa. If for all, I don't know how important this research on dissociated memories was around the world, but in Europe in the 19th, 20th century, during the time the research of Pierre Janet and Sigmund Freud were one of the most prominent ideas about dealing with repressed and suppressed memories. And actually, what they recognized was that many, many traumatic memories cannot be recalled as a matter of recall deficit and not as a problem of memory storage deficit. So they actually tried to find many ways to get to the memory in order to try to resolve many, many symptoms that such memories leave, including very severe depression. So I thought it was interesting that nowadays we actually can study even such memories using modern tools. Lastly, we can also use models of neurological disorders to study psychiatric phenomena. And you may wonder why I'm bringing these diseases. The reason why I bring them is that many neurodegenerative diseases actually start with anxiety and depression. So anxiety and depression typically occur in adolescence, but if we see it happen for the first time during adulthood, they can be some of the first signs of neurodegenerative disorders. And we have not explored this enough in animal models of neurodegenerative disorders, but there is more and more research in this field now. The reason for that is if we find early symptoms preceding neurodegenerations, then the efficacy and our options for prevention or delay of neurodegenerations, our chances to act in this very early phase and be successful would be much, much higher than to wait for the first symptoms of neurodegenerative deficits. This requires obviously the need to continuously test animals over a long time because these models develop over months. But we now have the options to actually do that, and not only to test the animals behaviorally, but also to do continuous measurements of all kinds of brain activities as well. And I will stop here now. I hope that with this lecture, I made some useful introduction for the other speakers who are coming up and who will be talking about many other molecular, circuit, and cellular mechanisms of various behavior in more depth and detail.
Ma'am, I have a question here. As you
Yes,
told
please.
about the anhedonia test of depression, it was shown that the rats were having sucrose preference. So is it possible also to correct? It is correct to say that in the people who are depressed, they have a preference for sugars. They have more preference for sugar.
Well, actually, it can happen. It can happen. But what the field now does, so what they assume is it doesn't really matter what the specific thing is. It's the change. It's whatever it is that individual find pleasure in, whether it is interest, hobby, food, social interaction, sex, anything. If this is changing and suddenly that pleasure is gone, this is actually used as an indicator of anhedonia in humans. So in some subset of patients, it will be, for example, chocolate. They stop eating chocolate. That's what parents come very often with adolescents. They would come and now they don't want to go out anymore with their friends. They stop doing this. They even stop eating chocolate. So some analogies are there in humans too. Not in all though, but in some.
Thank you.
Madam, really a very informative session. We wish that you could have talked for a little more on the other aspects as well. I have got two queries. The first one is, you're talking that there exists a relationship between oxytocin and autism. And generally nowadays, it has been observed that a good majority of children are now being born by cesarean section or C-section. And when and a newborn baby are delivered by C-section or cesarean, then there is very less release of oxytocin. Is it possible that nowadays the children which are being delivered by cesarean section, they might be suffering from, or there is a probability of suffering from autism?
Honestly, I don't think that I think that this has been largely dismissed. Somehow there was a lot of hope that targeting the oxytocin system, whether during birth and delivery, of course, when the oxytocin system is particularly active, or after birth, even during development, there were many clinical trials trying to balance and regulate the oxytocin release in order to prevent or reduce the severity of autism symptoms, but they have not been successful. So the oxytocin system doesn't really seem to play a key role there. There is much more emphasis now on these different proteins which organize the development of the brain, which are guiding the growth and connectivity in these most early stages of development, which are now the focus of research. Much more than oxytocin. So the idea is that the key changes occur much before birth.
Madam, there is one more question, that during accident or any trauma, when a person becomes unconscious, is it a dissociation state that you're talking about? And number two, whether it is beneficial for the overall health of an individual? Because generally, whenever any person meets an accident and the time period, again, when the person regains the consciousness, generally we don't, anybody, for example, I went an accident and generally I don't remember what happened in between that period. So there is, it's a zero state, neither there is any behavior, there is no memory of what happened in between. So is it a condition of dissociated state and it is beneficial for the health of an individual?
Actually, yes. So dissociation is viewed, the acute dissociation, so is believed to develop as actually a beneficial, as a defensive reaction, so that the person who is overwhelmed doesn't have to cope with overwhelming emotional and stressful events. So it is really seen as a strategy of the brain to delay or postpone resolving a stressful situation. So what usually happens though, and this is really a very puzzling phenomenon, typically there is really the dissociation causes the memories of that event not to be accessible, right? And you can see how different patients progress through their therapy. Some of them never want to go back and remember it. Others dedicate their life trying to remember it, that they know that something is there and that some pieces of memory are surfacing and are causing a lot of anxiety and depression symptoms. And for these patients, it's critical to try to restore the memory. So it really comes down to individual and their readiness to face their traumatic events. For some of them, it would be debilitating. For others, it's therapeutic. And this is really a huge problem for therapists to figure out which approach to take with different patients. But interestingly enough, it seems that the patients know.
Thank you, ma'am.
The short answer is yes, this is a very beneficial phenomenon in the short term. The problems don't occur in the long term.
Thank you, ma'am.
Ma'am, actually, I have one question. Am I audible, ma'am?
Yes, Deb.
Am I audible? Yes. Okay. Actually, we have already behavioral experiments caused stress on animals. So, the enzyme and the hormonal activity may be altered during that particular period. So, ma'am, my question is, what is the perfect time to sacrifice animals after performing any kind of behavioral experiments?
There is never a perfect time, you know. We always have to perform a series of tests in order to show how persistent and how long the effects are. Unfortunately, in the beginning, most stress effects were looked on in a very short term, in a very acute phase, within half an hour. One of the reasons is, as I mentioned, is because it is very unreliable to repeat these tests. But if you use the effects of stress on cognition, for example, then you can look at effects of stress over months. And I think that is much more reliable because you can see how long the effects last, when they start, when they stop, when the animals recover, when the animals worsen, whether they just stay stable. Because, you know, for individual animals, the time course will be different. And this is why it is much better to go for repeated assessment and repeated testing over a period of time rather than trying to choose the perfect time, because the variability there is quite large.
Thank you so much, ma'am. Great to see you after a long time.
You too, Deb.
I have one question. May I?
Yes, of course.
Madam, do environmental response change animal behavior? And if it is, what is the validate biomarker to evaluate acute and chronic response?
Yeah, so we have several different biomarkers that could differentiate those two. For example, acute stress response has a pattern of hormonal changes that we can get very, very easily. Those include corticosterone levels, glucose levels, insulin levels. So they're actually a range of very simple biochemical markers which we have known for a long time that can be very reliably used to determine the stress response on the acute scale. In the long term, actually, the simplest way to establish chronic stress is what Sally, who described stress originally, found. After a month of stress, the two things that one can see is immediately a hypertrophy of the adrenal gland, So you can see that the adrenal glands become huge, and that's really the best marker that we still have on chronic stress. And it's a bit paradoxical because we have measured all kinds of mRNAs and microRNAs and circular RNAs, and still this enlarged adrenal gland is still the best indicator of chronic stress. But we can also, in animals, for example, it again depends on the species. One of the strong indicators of chronic stress in rodents, for example, is they have skin changes. They start scratching, they have alopecia. So the skin is a very good indicator of chronic stress in rodents. And behaviorally, well, okay, I should stop with the behavior because you asked me for biomarkers. We can also see, for example, constantly increased levels of steroid hormones, but even more importantly, they have disrupted circadian rhythm. So usually many of the neuroendocrine systems in the body fluctuate regularly. And all these fluctuations, which also are very beneficial and important for sleep, recovery, and so on, these rhythms become disrupted.
Okay, thank you, madam. But one more question is here. Suppose there is an acute response and we are giving a acute environmental stress to our animal. And at the same time, the stimulus is chronic. So should we take a different marker to evaluate their responses?
You mean if we have at the same time chronic and acute stress?
Yeah, exactly.
Well, it doesn't, what we would do in that situation, the key thing is that we have all the adequate control, because we would need to have a group which is acutely stressed, a group which is chronically stressed, and a group that has both. And only then we can actually investigate which are the individual effects of acute or chronic stress, and then how their interaction happens. And very often, acute stress acting on chronic stress will give a very different quality of a response. So there are many labs, for example, that study now a combination of juvenile stress experienced in adolescence or chronic stress experienced with adolescence with acute stress in adulthood. And these stressors seem to be very severe, and their consequences seem to be very long-lasting. So they can cause very long-lasting behavioral changes which are consistent with anxiety or depression.
Okay, thank you. Thank you so much.
I have Malvika. Malvika, can you hear me?
Can we imagine a knockout mice with the ACTH-RH gene, and the animal will be stress-free, or there will be some other problems?
I'm sorry, I didn't hear very well. I hear some echo now.
I'm asking, can
I lost the sound. Sorry. Oh.
Thank you very much, Dr. Jelena, for a very nice and informative lecture. And may I now...
Thank you, Professor Jelena. I also thank you.
Thank you, Janak.
Thank you.
Good to see you.
Yeah.
Is that Raj Kumar there behind us?
I'm
But
sorry.
Yes, ma'am, it's me.
It took me a while to recognize.
So thank you once again. And now I would like to invite the second speaker of this session, Professor Sukala Prasad. He is from the Department of Zoology, Banaras Hindu University, Varanasi, India. Professor Sukala Prasad completed his graduate and master's studies in zoology with biochemistry as a major elective course from Department of Zoology, Banaras Hindu University. Thereafter, he obtained his PhD degree in zoology on age-associated studies on HMG proteins of the rat liver from the same department. Later, he joined Department of Zoology as assistant professor in Department of Zoology in 1991, and currently he is serving the department as a professor since 2008. He visited National Cancer Research Institute, Tokyo in
इनको और मैसेज में करो
I'm audible.
No.
I think this is time to start the presentation. Some audio interruption is there. Sound voice is not coming. Am I audible?
May I now request Professor Sukala Prasad to start his
Thank you very much, respected chairpersons, to invite me for presenting today's morning talk from my side. At the outset, I must thank Professor Janak Dulari Ahi and Dr. Rajkumar Koiri to enthuse me to get associated with you and to be part of this international webinar being organized at Department of Zoology, Sagar University. Dr. Rajkumar Koiri and I see that Dr. Rasmi Srivastav, they have been our proud product and they are serving Department of Zoology in Sagar University. I feel proud of them. They must be contributing their best there, I hope so. And what I am going to talk about, let me share. Am I audible as well as my slides are visible?
Yes, sir. Yes.
Okay. And can you see the full slide?
Yes, sir.
Okay. So I thank the organizers again to invite me for delivering this talk at very important international webinar organized by Professor Ahi and team. As Professor Jelena talked about various animal models of various conditions like neurological disorders, I will talk about how brain function, especially in the light of learning, memory, and related behaviors, what we call as the cognitive behavior, that is affected by a kind of pathophysiological condition that is called hypoxia. So the title of my talk today is Molecular and Neurobiological Basis of Hypoxia-Induced Cognitive Impairment and Neuroprotective Effects of Bacopa monnieri extract. And for understanding this, in
Sir,
fact,
sorry
we are going,
for sir, sorry for interruption. Please make your slide full screen. It's
is
not
it
full
not full?
screen.
But I have, from my side, I have made it full screen. Is it not visible there?
Sir, it's visible, but it's not in full screen mode. Sir, no issue. You go ahead.
Okay, okay. So cognitive behavior is one of the functions of the brain. Let me mention few of the factors that affect in general the brain function. There are several parameters, anatomical, physiological or pathological or metabolic disorders. Okay, so if you look at the pathophysiological conditions, the most important thing that affects the brain function is aging or related neurodegenerative disorders, either only because of aging or during aging the brain has undergone some disorder that is because of the diseases. And these diseases are not the mark of aging, but aging leads to onset of several conditions, neurological disorders. Hypoxia is one of them. Hypoxia is one of the major denominators of the above changes in the brain. On the other hand, I will not spend a lot of time. The metabolic, including diabetes mellitus, hypercholesteremia, deficiency of omega-3 fatty acid, thyroid disorder, or vitamin deficiency. Many parameters are there that affect the brain function. And this is what is a summarized slide, more focusing towards hypoxia. You know, the brain consumes the maximum oxygen, being only.
Sorry to interrupt you. Your slides are not moving.
Slides are not moving.
Yes, sir. So sorry to interrupt you.
No problem, no problem.
No, it's okay. Sir, now it's okay.
Now it's okay. So I am focusing now. I have talked about the brain function, what are different factors. The previous slide had shown. This is in gist, again repetition of the same thing, but let us focus on hypoxia.
Sir, I would request you, sir, I would request you to increase the percentage of your slide since it's not in full screen. From the bottom, if you can increase the percentage from 70 to 80 or 90, I think it will. Sir, you can once you can disconnect the full screen mode and again you can reshare it. I think the full screen problem will be sorted out. If you can once you can stop sharing and again you can start sharing again, I think then it will come into the full screen mode. Meanwhile, it's not coming in the full screen mode.
Okay, so I disconnect myself.
So don't disconnect yourself. You just remove the full screen mode, stop sharing, and then again start resharing it, and then F5. I think it will come into the full screen mode.
So I am stopping sharing.
So sorry to interrupt you again.
No problem. You are telling for the betterment. So now I'll have, as you are suggesting, this is not in slide mode. Okay.
Yes.
What I have set on my screen, this is not in slide mode, and this is in 70% of the slide size. Okay.
So now you can reshare it. I think the problem will be sorted out.
Okay. So
Reshare it and press F5. That problem will be sorted out, I think.
okay, I will reshare it. Still on the left hand side, you must be seeing the panel.
Yes, sir.
Now, can you see the full slide?
Sir, still not in. Sir, click on the full screen mode or press F5. Press F5.
Full screen mode. Okay, full screen mode. That I have done. That I have done, but I am not seeing the presentation.
Sir, it's okay. You can continue, sir. Presentation is coming.
Presentation is coming.
Yeah, we can see your presentation.
But I am not seeing my presentation in this condition. Are you able to see the presentation?
Yes, sir, we can see the presentation.
And are you able to see the
Yes, sir, slide is moving. Slide is moving now.
slide is moving now?
Yes, sir.
Shall I go back?
Yes, sir.
Okay, so this part I had explained. Okay. Now I was at this state of time. Is it visible now in full slide mode?
Sir, you can continue. No issue, sir. Although it's not visible in full screen mode, but you can continue. We can read the slide.
Okay, okay. So if you go through, there are several factors that may lead to hypoxia. This may be because of the disturbance at the during embryonic development, or it may be because of the respiratory distress during parturition of a child or baby, the persons traveling to high altitude, or inflammation, cardiovascular diseases, asthma, cancer, or traumatic brain injury, and of late the big problem, pandemic of last 2 years, including the present year, the COVID-19, that has led to the acute lung infection, severe lung infection, and supply of deficient oxygen to the body. So that has been on the focus. That is why I have marked it as very important point. So there are the several causes, and now if you look at the symptoms of the hypoxia, the person starts rapid breathing. There is a state of cyanosis over the body, poor motor function, and mental or muscle fatigue. Altogether, the person may also suffer from learning, memory, and cognitive deficit. Okay, so this is all because of the poor supply of hypoxia. The normal supply of the oxygen to the body is, in physiological condition, more than 20%, 20.9% of the oxygen in terms of the partial pressure. this is what is the normal condition. Now, if you look at how the hypoxic condition is maintained and how does it bring its effect compared to what does not happen in normal condition when the normal amount of oxygen is supplied to the body. In that state, when the oxygen supply is normal, there is a hypoxia inducible factor. The story starts from here. And the state of condition when the normal oxygen supply is there, that is called normexia. Okay. So in this condition, the HIF1 alpha, which is a transcription factor, that is finally degraded. Okay. So what happens when there is normexia? In presence of an enzyme called prolyl hydroxylase, when the 2-oxoglutarate is converted into succinate and hydroxyl moiety that is attached with the HIF1 alpha. Okay, and this invites or attracts several cofactors. One is 1-hypyl-Lingdau protein factor. Okay, so this binds to it, and once this binds to the HIF1 alpha, the proteasomal degradation depending on ubiquitinization in presence of E3 ubiquitin ligase complex, the HIF1 is degraded. So this frees the body to utilize the oxygen, or oxygen, normal supply of oxygen does not allow elevation of HIF1 alpha, which is state of the mark of hypoxia, that is hypoxia marker. If oxygen supply is less, then in that condition, hypoxic condition, HIF1 alpha is bound to another isoform or another subunit that is called HIF1 beta. So HIF1 alpha beta complex is formed and it binds to several other factors. Ultimately, it binds to hypoxia response element on the promoter of several genes, and that is how the state of hypoxia goes on, and hypoxia induced several things now start taking place biochemically. So during hypoxia, expression of HIF1 is upregulated, which is made up of, it consists of constitutively expressed HIF1 beta subunit and oxygen tension regulated HIF1 alpha subunit. And we have worked on animal models, so there was a requirement of developing the hypoxia model. There are several ways of developing hypoxia models. One can use physiological modeling system using hypoxia chamber, where one can deliver known amount of or known tension of the oxygen mixed with nitrogen, or chemicals like deperoximine and or cobalt chloride. Okay, cobalt chloride is chiefly used as one of the methods. So how does cobalt chloride helps in developing the model? So I can say here, if this is hypoxia condition, then this binding of 1-hypyl-Lingdau protein, which is ultimately going to destroy HIF1 alpha, this process is inhibited by state of hypoxia. Certainly, then cobalt chloride must be doing something like hypoxia does over here. So in the next slide, this is what is cobalt chloride does. Cobalt chloride does the same thing what is physiologically done by hypoxia. It binds to iron, chelates with iron, and this does not allow HIF1 alpha to bind with VHL, 1-hypyl-Lingdau protein factor. Okay, so once this is HIF1 alpha is not hydroxylated, it is not allowed to undergo degradation or the proteolysis. So this state is created. Now, and how this happens, the cobalt chloride inhibits the enzyme prolyl hydroxylase. Now, there are several studies that has been carried out. What happens in case of the hypoxia? Severe and prolonged hypoxia contributes to brain damage mainly because of neuronal dysfunction and cell death. This is one of the findings. And several studies have shown that injury to the brain due to less supply of oxygen triggers impairment of learning and memory deficit. Okay, and hippocampus in particular, CA1 or CA3 region has been reported to be severely affected by hypoxia. So this is all in the literature, but the clear-cut mechanism or the biochemical or molecular basis is not clearly established. So this is again a kind of what happens during course of learning associated function of the brain, how hypoxia leads to. This leads to neuronal damage, synaptic transmission of glutamatergic, GABAergic, cholinergic neurons. It also leads to oxidative stress, leading ultimately behavioral impairment. And this is all because of, if you look at the synaptic function, the synaptic arrest of neuronal circuit that takes place. And post-synaptic efficacy that also is affected. So this is what is already in the literature. What we have done, now we can see what is the state of situation while forming the memory. You can look at the brain. I think all of you are already known about different parts of the brain. The temporal cortex is the structure which is associated with the development of learning and memory, especially long-term memory. This is hippocampus.
Sir, sorry to disturb you. Slides are not moving.
Slides are not moving, but I see it is moving in my system.
Sir, on the top it is showing resume slide. So, sir, if you will click on the top, you can see on cobalt chloride it has been mentioned resume slide. So if you will click on resume slide, so it will start. Resume slideshow.
Where it is?
Sir, on our screen we can see at the top of cobalt chloride there is a icon on at top of cobalt chloride where it is showing resume slideshow. Still we are on slide 4. Sir, you can stop sharing the slide. I can and you can reshare the slide. Yeah, now it's okay, sir. Now it's okay. It's moving.
It's moving now.
Yeah, sir, it's moving now.
Okay, let me go ahead. Now, is it there?
Yes, sir.
Is
it's
it moving?
Yes, sir. It's moving now.
So what I was telling that we have to. I mean, let us see where is. You know, hippocampus is the site where long-term memory is consolidated. So this is the temporal cortex, in addition to several other regions of the cerebral cortex. It is the temporal cortex, okay, that houses the hippocampus for the long-term memory, is the seat of learning, memory, and cognition. In the hippocampus, there are several detailed structures in which there is a structure like that is called dentate gyrus, which communicates with the region of the hippocampus called CA. CA term has come from Cornu Ammonis, the Greek god of knowledge. In our system, in our mythological system, we have goddess of knowledge, Saraswati. So Cornu Ammonis was a Greek god of the knowledge. So dentate gyrus communicates to the CA, Cornu Ammonis. It has different areas. So dentate gyrus first communicates with mossy fibers to the CA3 region. Then it goes to finally CA1 region, and ultimately the information is stored in the subiculum which is ultimately called as hippocampal formation. So that is there, and all these things, when the dentate gyrus is communicating to the CA region, this all takes place while there is the intra or interneuronal network or crosstalk between one neuron that makes the dentate gyrus or that makes the cornu ammonis. Okay, so this happens by neuron crosstalk, and this takes place on the dendritic spines. So the cell soma of the neuron has dendrites, and dendrite has several button-like structure, mushrooming structure, and this structure communicate to rest other side of the neurons, like here the neurons in the dentate gyrus or neuron in the CA area. Okay, they synapse with each other, and then the discussion goes on in terms of release of a chemical called neurotransmitter. There are several neurotransmitters. They are relevant for the cognitive function of the brain: glutamatergic system, cholinergic system, dopaminergic system, GABAergic system. In varieties of ways, they crosstalk. None of the system is alone is responsible for completing the task of cognition, but for the experiment point of view, for the work point of view, which is a part of a piece of work which is I am going to present over here, is based on the finding on how glutamatergic system is altered during hypoxic condition, and the hypoxia model was made accordingly. Uh, so, let me see what is the glutamatergic system. So this is what I was telling. This is CA3 neuron, and this is CA1 neuron. We have to imagine like that. The final discussion or crosstalk is between CA3 and CA1 after the information has come from rest of the brain by perforant fibers to dentate gyrus, and from dentate gyrus it has gone to the CA3. So CA3, once it is stimulated, that means this neuron in action dependent or activity dependent manner, it releases glutamate, and glutamate binds first to the gateway of the postsynaptic neuron, gateway of the cognition, that is learning, memory, and these are the glutamate receptor type 1, which is called EMPA receptors. Okay, this is pharmacologically a type of the glutamate receptor which is excited by EMPA. That is why it is called EMPA receptor, otherwise it is a glutamate receptor. So once the CA3 neurons are activated, so Rajkumar, if I don't make it on slideshow, the movement in the internal movement in the slide will not be possible. So can I go through that?
Yes, sir. No issue, sir.
Okay. So what I am seeing here, the glutamate now that binds with EMPA receptor, and once it binds to the EMPA receptor, the EMPA receptor conformational change takes place that permits entry of the binding of the glutamate. Post binding, the cytosolic sodium level increases, and because of that the postsynaptic membrane undergoes depolarization, and this is a kind of signal to another type of the glutamate receptor. This is called NMDA receptor. So glutamate binding here leads to the entry of calcium ion along with sodium ion by removal of the magnesium ion block. And once the calcium level increases, the calcium binds to the calmodulin, which ultimately activates cascade of reaction, and this is what is mentioned over here. And then after that, there is one more type of glutamate receptor, what is called as metabotropic glutamate receptor. So that also is activated. So the activation of CA1 neuron is not only by mPA receptor, it is in combination with mPA receptor, NMDA receptor, and metabotropic glutamate receptor. Okay, and once metabotropic glutamate receptor is activated, it increases the mRNA translation, including mPA receptor and other receptors. And there are proteins which can inhibit or which can suppress the mRNA translation, and varieties of other things also takes place in presence of excess of calcium, that is activation of reactive oxygen species and leading to neuronal death. So this is all the story, and once the role of glutamate with the binding of mPA receptor first and then binding with other two receptors of glutamate, the glutamate is taken up by the neuroglia, that is here astrocytes, through excitatory amino acid transporter called GLT1, and glutamate ultimately is converted into glutamine, and glutamine is recycled to the presynaptic neuron. Here it is CA3 neuron, and glutamine is then converted to glutamate. So this is what is recycling of glutamate. In addition to the new glutamate is being formed. So now let me see how this. I am focusing on how mPA receptor, and later on I will tell about how metabotropic glutamate receptors, their expression is affected by state of hypoxia. Okay, so sorry. Now, as I was telling, mPA receptor expression is one thing. This remains on the membrane. It is expressed on the membrane, but it is maintained in this state so that it does its function. It is maintained by several scaffolding or trafficking proteins. A few of them I am mentioning. There is a protein called TARP, and especially TARP gamma 2, also called as stargazin. In presence of PSD95, the mPA receptor is stabilized over here. For that, the GLUR2 subunit of mPA receptor, they need to be phosphorylated. Okay, and once it is phosphorylated, hyperphosphorylated, it depends on which region is being phosphorylated. There is another trafficking protein called PIC1. Okay, so PIC1 is responsible for the TARP and PSD95. You have seen the PIC1 is responsible for endocytosis. That means removal of the glutamate receptor, and it is now, if mPA receptor is less, the state of learning and memory developed will be poor. If it is little more, it is indicative of that postsynaptic potential is more, which is required for the learning and development. If it is so much
Sir,
high,
slide is not moving.
I am not moving. I am
Okay, fine.
maintaining the same slide. Okay.
Okay, sir.
So I am telling about. Let me go back to previous slide. Yeah. So the amount of or the level of mPA receptor is a kind of mark. Okay. If there is too little of mPA receptors expressed on the membrane, there is poor cognitive development or learning or memory development. If the memory has to develop, the number of mPA receptors has to increase, or it increases. This has been reported. But if it is overexpressed, then it becomes neurotoxic or excitotoxic. There is a term called excitotoxicity, that is there. Much have been talked about excitotoxicity in terms of NMDA receptor, but the same story can be there because of the overexpression or huge expression of EMPA receptors. So what is the message here? That EMPA receptor has in one state has to increase for the better function of the learning and memory, brain function. If it is overexpressed, then it will lead to excitotoxicity via the excitotoxicity induced by overactivation of NMDA receptor. So there is a direct relation between the activity of EMPA receptor and activity of the NMDA receptor. High activity of or large activity of the EMPA receptor is proportional to the increased activity of the NMDA receptor, therefore the excitotoxicity. So a state of excitotoxicity is a kind of a state that leads to the LTD, long-term depression state. You know, the LTP and LTD, these are two states of electrophysiological or physiological, biophysical aspects of the learning and memory. This plasticity has to be maintained with dynamic equilibrium. And once this is maintained, then the learning memory development is normal. Is my slide moving now?
Sir, not yet. Still, we are on the first slide that you started 10 to 15 minutes before. Now it's moving. So now it's moving.
Okay.
So now it's moving. Now we can see the AMPA receptor slide. AMPA
Okay,
receptor
so
trafficking pathway.
yeah. So this I had described. So I was telling that if the TARP PhD 95, if they are undergoing change, it is going to affect the expression of the EMPA receptor on the membrane. So more of the TARP, more of the PhD 95, that will be associated with more of the EMPA receptor. And if the peak 1 level, if it goes up, that means it will withdraw EMPA receptor from the membrane to the cytosol, and this will lead to the memory dysfunction, or you can say the decline in the memory. So this is what is the state of situation, that the not only the EMPA receptor but the trafficking protein or scaffolding protein, that is also required to be maintained in the in proportion to the glutamate receptor. Okay, to maintain the normal level of EMPA receptor, the normal expression of TARP and PhD 95, that has to happen, and the peak 1 has to be maintained at low level, so that the EMPA receptor is maintained on the postsynaptic membrane. If this increases, that means the TARP and PhD 95 will not be able to scaffold the EMPA receptor on the membrane. So this is what is the state of situation. If any alteration takes place, that will be in respect to the functioning of the EMPA receptor. Okay, so now other aspect which I would like to discuss, there are so many herbal preparations or the herbal drugs. One of them is Bacopa monnieri. In Hindi or in the Ayurvedic literature, this is called Brahmi, the memory booster. This has been shown to boost the memory in normal conditions. What we have done, we have checked if hypoxia leads to memory dysfunction or cognitive impairment, then how much Brahmi is useful getting back to the state of normal cognitive function of the brain. So we hypothesize that glutamate receptor expression and trafficking, this play crucial role in consolidation of memory, and it may be altered by hypoxic conditions leading to memory impairment, and Bacopa extract may play neuroprotective effects by modulating above. This is what is our assumption, our hypothesis, and with that we asked few questions. One of the major question was whether hypoxia induced spatial memory impairment is associated with alterations mainly in EMPA glutamate receptor with its trafficking proteins metabotropic glutamate receptor R/5. This is group 2 metabotropic glutamate receptor, uh, metabotropic glutamate receptor R1, R5. They are grouped together, so they are called GLU-R1, GLU-5, M-GLU-R1/5. Okay, a study has been made with M-GLU-R1 or 5, and the Bacopa monnieri extract, which is shortly written as BME, reverses this alteration. This was the major question. And whether the hypoxia-induced memory impairment is associated with alteration in neuronal cell density, dendritic spine density, and can this effect may be reversed by Bacopa monnieri extract. So these were two primary questions we were supposed to answer first. So studies were carried out as follows. We first developed the hypoxia model and we checked whether the animal is suffering from hypoxia using the molecular validation, effects of hypoxia on the spatial memory, the behavioral validation, and then we studied the effect of hypoxia-induced memory impairment and its association with alteration in expression of EMPA receptor and its trafficking proteins and metabotropic glutamate receptor 1/5. And then we saw the effect of BME on above alterations. The work plan was the animal, one of the group of animal that was maintained as control, and then hypoxia was generated using cobalt chloride, and by pilot experiment we came across that 10 mg per kg body weight cobalt chloride is sufficient to induce hypoxia, and we then used several other concentrations and then established ultimately what is the suitable concentration of or dose of the cobalt chloride. We came out and then we isolated hippocampus, and then we studied the expression of hypoxia markers, the biochemical markers, HIF1 alpha and many others, and data were analyzed. So let us first see what is the effect of dose-dependent treatment of cobalt chloride. With this, you can see that in comparison to control, the 40 mg of cobalt chloride was able to induce optimal state of hypoxia, and that is measured at the level of the alteration in the HIF1 alpha level. Okay. So other higher concentrations did not lead to the increase in the expression of HIF1 alpha. Why this is so? This is again a question to be answered, and the work is going on. But for the moment, since we observed 40 mg as the optimal concentration of the cobalt chloride, we started with this for developing rest of the work. So using that, we checked. Let me maximize here. I'll go back then. We found that in case of hypoxia, as you saw earlier, HIF1 alpha, its expression at both RNA and protein level by semi-quantitative RT-PCR and by Western blotting, we saw that there is overexpression of HIF1 alpha. That validates the hypoxia model. The other markers were also checked. This is called glucose transporter, that is GLUT1. So its level also is increasing. That is another way to validate the effect of hypoxia. There is other marker that is called erythropoietin. In case of hypoxic condition, erythropoietin level increases to maintain the number of RBC, therefore bringing back to the state of normal condition of oxygen. So erythropoietin level increased at both RNA and protein level. So altogether, this suggests that the hypoxia model we created, that was a model. This work was published long back. So this validated our model. Now we went to the actual planning using the uh, uh, pilot experiment led optimal concentration of the cobalt chloride. We had a control, and then we also had Brahmi, the control treated with Brahmi, and we had two states of hypoxia. One is hypoxia with 15 days treatment of cobalt chloride, and then the the animal was sacrificed at 8 days after hypoxia, and another group of animal, they were sacrificed after one more week. That means altogether 15 days of hypoxia. So 8 days and 15 days. And then we saw the effect of BME, hypoxia treated mice. Before that, the Bacopa treatment was given, and there was a group of animal, the hypoxia was generated first and then Bacopa was given. It is just to see whether the Bacopa has a neuroprotective role or it has a neurotherapeutic role. Ultimately, both are neuroprotective role, but we wanted to see before and after effect of the Bacopa. Once the hypoxia is generated or Bacopa is given before hypoxia is created. So the spatial memory using Morris water maze test, that was established, that was studied, analyzed, and thereafter the biochemical or molecular biological experiments were carried out and data were analyzed. We first checked what is the effect on the HIF1 alpha. Okay, we saw that the hypoxic condition 8 days or 15 days. Okay, both the conditions HIF1 alpha is overexpressed at RNA as well as at protein level, and the Brahmi pre and post treatment both significantly brought down the expression of hypoxia 1 alpha, HIF1 alpha. Okay, that means Brahmi is regulating, or Bacopa is Bacopa extract, which is rich in bacosides A and B. This is in the literature. So that decreases the level of, or it downregulates the level of HIF1 alpha, which was upregulated in case of hypoxic condition in both the situations, the prolonged hypoxia and the 8 days hypoxia. Okay, so here it is a good correlation that Bacopa has a favorable role. Okay, now this is what is the Morris water maze test paradigm for the analysis of a spatial memory. I don't have to tell much about it. I think all of you, they are well familiar with this test. So mice initially they are trained, they are allowed to swim and search a hidden platform in the water. Okay, and slowly they learn and they take less time to reach to the platform. This is the actual state. I am not showing the video, but this is the hidden platform, and animal learns how quickly or how early to reach. If they are learning better, they will take less time, they will cover less distance. So the time taken and the distance covered, these two are reduced. So if this is so, then it will be said that animal has developed spatial memory. Spatial memory because the platform has to be located. Where is the platform? So that is why it is called spatial memory. Now let us see what is the result In control, after learning, this is what is the animal after training. This is the time taken, or there are several latency time is there, the distance covered, path length that is traveled, that is there, number of platform, let me increase the size for me at least, the number of platform crossing and time spent in the platform quadrant. Okay, so based on this, ultimately the conclusion has to be drawn. The Brahmi treated animal as the track pathway. This is video tracking record. The time consumed over here is reduced. Okay, this is for the time, and also the tracing also says that the hypoxic condition, the animal is taking lot of time and it is traveling so much for searching the hidden platform. But Bacopa treatment, in both the cases you see, the Bacopa treatment reverses the time spent to search the platform or the distance traveled to search the platform. Okay, so on various scale and the latency scale, you can see there is a significant decline in the time required for reaching the platform, okay, which was adversely affected by hypoxia under both the conditions, the early conditions, the late conditions. Okay, and this is the path length, that also says the similar thing as was latency telling, and this is the number of platform crossing. Platform crossing increases with the help of the Bacopa treatment, which was not found in case of hypoxia. Okay, that means they are unable to retrieve their memory to reach to the platform. Bacopa was improving this situation significantly. Okay, and the time spent also increased in the platform quadrant for in absence of the platform, okay, compared to the hypoxic condition. So behaviorally, it is clear that hypoxia alters the memory. So this is behavioral validation of the effect of hypoxia on cognition. Now we come to the glutamate receptor level. What was observed? The hypoxia 8 days and 15 days. Okay, this is the state that at RNA at protein level, one of the subunits of glutamate receptor, which is very important for the viewpoint of allowing sodium into the cell during course of glutamate receptor activation, EMPA receptor activation, the GLUR2 is very important, and its level in both the conditions of the hypoxia compared to control, that is this is true in case of RNA as well as protein. So level of glutamate receptor 2 subunit, that is significantly increased. Of course, there is time dependent upregulation of GLUR2, but in general we can say that compared to control, the amount of glutamate receptor that is increasing so much. But the pre-treatment of hypoxia, not the after treatment, pre-treatment with the Bacopa, that significantly decreases the expression of the GLUR2. Okay, so it seems that compared to control, the hypoxic condition, especially the long-term hypoxic condition, it excessively upregulates the level of GLUR2 in the hippocampus. Okay, so that may be a cause of over activation of the EMPA receptor and the excitoneurotoxicity caused by the overaction of the overall EMPA receptor. So that is assumption. We have not checked that way. Now let us see what happens in region-wise structure of the hippocampus. So firstly, the level of GLUR2 in CA3 region. So this is with the immunohistochemical work. The coronal section of the brain was made and the hippocampus region was focused, and it was in CA3 region. It was found that hypoxic condition, as was seen in case earlier Western blotting system, and the RT-PCR system, the
एक मिनट ना
GLUR2 expression is overexpressed in this region also. Pre-treatment of Bacopa extract that downregulates
ठीक
the overexpressed GLUR2 subunit. Okay, so this was found in case of CA3. What happened in case of CA1? Okay, in CA1 again there was overexpression of the GLUR2 compared to control. There was, so one can see here the Brahmi also was increasing, but this increase is of course significant, but not like hypoxia is increasing the GLUR2 level in CA3 as well as in CA1. Okay, in the Western also Brahmi was doing something, but the extent of upregulation is very much high in case of hypoxia compared to only Brahmi what was doing. Okay, so this may be a favorable overexpression. This may lead to the unfavorable situation that may lead to the loss of memory. And Brahmi here was able to significantly reduce the level of GLUR2 expression in CA1 region. So within the hippocampus region wise, there is overexpression of GLUR2. There is
चलिए
background
दिखाइए
noise. Rajkumar, can you
Bhagwati Sahu, please mute your mic. Bhagwati Sahu. Bhagwati Sahu, please mute your mic.
So okay. So Bacopa is significantly downregulating the level of GLUR2, okay, compared to hypoxic condition. But the same one with later treatment with Bacopa, it was not that much protective. Okay, so here is difference between pre and post treatment with Bacopa. Now let me see what happens to scaffolding protein. So far it has been proven that there is hypoxia-led overexpression of GLUR2, or you can say in general EMPA receptor, which is taken care of by pre-treatment of Bacopa monnieri extract. Now since there is overexpression of the EMPA receptor due to hypoxia, we checked what is the state of effect or state of condition with PSD 95, which is known for maintaining the concentration of EMPA receptor on the membrane, as I had explained earlier. We observed that at protein level, hypoxia has no significant effect on the level of PSD 95. Okay, but in case of RNA, PSD 95 is significantly decreased. Okay, the RNA for the PSD 95 is decreased, but protein is unaffected. So it indicates that finally the role is played by protein. Protein is unaltered, that means PSD 95 is not the factor which is affected. But what could be leading to change in the RNA? This is what we have observed as the steady state level of the PSD 95 RNA. This may be because of the high turnover of the RNA. That means more of the RNA is degraded than it is being synthesized, and at its steady state there is less RNA. So this could be exceptionally because of the RNA turnover mechanism but so far the steady state level of protein is concerned, there is no change in the protein. There is no change in the protein. So there is no direct correlation between transcription of PhD95 and the expression at the level of protein level of PhD95. So with this, we can say that PhD95 remains unaffected, but pre-treatment little bit enhances the PhD95. Of course, hypoxia does not do anything. So this could be one of the assumptions that Bacopa maintains the PhD95 as is does alone compared to the control. And that is what is observed in case of hypoxia. Lot of question may be there. We are still studying why this discrepancy in respect to hypoxia and also in respect to pre-treatment of Bacopa is there in case of PhD95. This is being studied. But so far, this was the finding. Now, let us see what happens in case of TARP. Once again, I remind the TARP, if the glutamate receptor has to be maintained at a particular expression level, TARP has to be maintained at that level. So we found that the hypoxic condition increases the expression, significantly increases the expression of TARP protein as well as TARP RNA. Okay. So compared to control, the level of the TARP in hypoxic condition, of course, the prolonged hypoxia increases the hyper expression or over expression of the TARP. This favors the over expression of EMPA receptors. So that is already, if you go back and remember, EMPA receptor level is increasing, supported by increased level of the TARP. Okay, so this goes in favor of the TARP and EMPA receptor. They are correlated with each other. More of the EMPA receptor, more of the TARP. So more of the TARP favors more of the EMPA receptor on the membrane, which is reversed, especially by pre-treatment with the Bacopa monnieri extract. Okay, and this reversal is not complete exactly up to the control level, but this goes towards the normal level. So reversal is not complete, but reversal is towards the normal control level. So this supposed to Bacopa treated animals, they remember better. I mean, their memory power is coming back. Where they have lost, they are gaining memory power. This is in favor of that line, that Bacopa treatment leads to lowering of the TARP expression. Therefore, that also is correlated with the lowering of the EMPA receptor GLU-R2 subunit. Now, let us see what is at the level of peak. I again remind that peak 1 is associated with decreased level of EMPA receptor on the membrane. Okay, so more of the peak, which is not, I mean, if you look at the protein level, the hypoxic condition, especially prolonged condition, significantly decreases peak 1. Okay, which goes in the favor of hypoxia-led overexpression of GLU-R2. Okay, so if more of the GLU-R2 is there, that is because of it has been in this work, it has not been included. For binding of peak 1, GLU-R2 phosphorylation is required. If you go through the previous slides, several previous slides, you will see that peak 1 binds only when GLU-R2 at certain point they are phosphorylated. So that phosphorylation has been reported to increase, and the peak 1, then expression increases, and more of the peak 1 will bind to more of the glutamate receptor, and that is why now the hypoxia, especially prolonged condition, that this is going to decrease. Okay, and Brahmi, it is going to upregulate uh, compared to this and compared to control. Okay. So, I mean, better we compare to hypoxic condition. So, these two, pre and post both, it maintains the level of peak 1, okay, which was not by hypoxia. Hypoxia was able to down-regulate the peak 1, and this is what is correlation between the GLU-R2 expression, and I am not showing the phosphorylated GLU-R2, which increases so much, and that is how it has been correlated. Now let me see what happens at metabotropic glutamate 1 or 5 receptor. Okay, we saw that hypoxia leads to decline in the level of the metabotropic glutamate receptor 1/5, okay, compared to control, and the pre-treatment especially upregulated the metabotropic glutamate receptor 1,5. Okay. So this is connected to the favorable role of Bacopa in maintaining the concentration or the level of or expression of the metabotropic glutamate receptor on the membrane towards normal control. And this was studied at the level of immunohistochemical level. The similar story was found in CA3. The metabotropic glutamate receptor expression in the early and the prolonged hypoxic condition, its level is decreasing, but Bacopa pre-treatment especially upregulates the uh, metabotropic glutamate receptor 1,5. Okay, so that is a significant change. Similar trend was found in case of the CA1 region of the hippocampus. CA1 region. Again, the hypoxic condition, especially the both are significant. The level of GLU-R1, GLU-R5, I mean metabotropic GLU-R1/5, they are downregulated, which was reversed by pre-treatment of Bacopa towards normal. Okay, so this, as I stated earlier, now we can summarize. Hypoxia leads to the overexpression of the GLU-R2, which may be associated with excitotoxicity, glutamate-based excitoneuratoxicity, and that is favored by alteration in the level of the glutamate receptor or EMPA receptor trafficking proteins so far. And this is by also decreasing the level of metabotropic glutamate receptor and reversing the metabotropic glutamate receptor level with respect to Bacopa monnieri extract. Okay. So we can summarize this part of the work. Decreased peak 1 and increased TARP may lead to increased GLU-R2 expression on the post-synaptic density, which may in turn induce neuronal excitotoxicity, leading to neurodegeneration in the hippocampus of hypoxic mice. And Bacopa monnieri extract pre-treatment restores above towards normal, which might be correlated with recovery of memory impairment. So this is what is the data they are suggesting us. And the expression of metabotropic glutamate receptor, as I said, it showed a significant decrease in hypoxic condition, which was reversed by Bacopa extract. Okay, decreased expression of metabotropic glutamate receptor 1/5 in hippocampus, especially in CA3 and CA1 region, may be responsible for impaired spatial memory due to hypoxic condition, which can be restored by administration of Bacopa extract to hypoxic mice. So this is what is so far we observed. Now since we are assuming that this may lead to neurodegeneration, that means decline in the active neuronal count. So we assumed the other aspect of the work, and then we planned accordingly in the same way. We, by perfusion experiment, we took out the whole brain and underwent the crescent violet staining. This is what is also called as Nissl staining and rapid Golgi staining, and the data were collected and analyzed. Now, this was done in the CA3 region as well as in CA1 region. The assumption was that hypoxia is leading to neurodegeneration. If neurodegeneration is there, that means the number of neurons or neuronal cell density that must be affected. So to know this, the software-based analysis was done after staining the sections, brain sections across the hippocampus, and then the cell density was analyzed. It was found that in case of the hypoxia, the neuronal density was significantly declined. So this is indicative of that number of active neurons, they are declining. It was found that these animals, when they are treated with Bacopa pre and post treatment as was done earlier, it was allowing more number of cells to survive. Okay, that means here is the neuroprotective role of Bacopa. The number of active cells or neuronal cell density went up. So I don't mean to say here the cells which died, they were they were recovered. In fact, the neuronal density may come back to the normal because of their neurogenesis in the hippocampus. It is well established that dentate gyrus neurons, through their neuronal stem cells, they get back the number of active neurons or live neurons, and they perform the similar role to gain the memory. Or so our data is going in that direction. We had some other work also to prove that these cells are dying because of apoptosis in other case of work. We are not presenting that. I am not presenting that data here, but this is indicative of that these cells are not that much dying, or cells are being regenerated. Regenerated means neurogenesis is taking place, not the dead cells are coming back and they are becoming live. Okay, this is not the kind of experiment we did, or neither we found that. We don't want to say that also. So we can only say that neurons were not allowed to die much enough, or neurons were regained because of the neurogenesis, whatever the mechanism might be. But we see that there is significant rise in neuronal cell density after Bacopa treatment, after the hypoxia was achieved. So before and after both state of Bacopa treatment increases the neuronal cell density. This is in case of the CA1 region. Okay, similar story. The neuron cell density that went up, okay, compared to both the state of hypoxia, the 8 days hypoxia and prolonged hypoxia, that is 15 days hypoxia in our experiment. Okay, and later on we also analyzed the dendritic spine density. Okay, we can see the dendritic arbor here. Okay, in case of hypoxia, the shrinkage of the dendrites can be observed compared to the control, and then the dendritic output arbor, this was found to be increased in case of the Bacopa treatment. Now, by amplification of this, going through Neurolocida software, the neuronal cell density that was counted Okay, and it was found that hypoxia leads to decline in the dendritic spine density, and the Bacopa treatment, pre and post both, they resulted into rise in the dendritic spine density. So this was the finding. This can be seen over here also, that the most of the mushrooming was done in case of when the Bacopa treatment was given. Okay, so this is clearly showing that the molecular finding is correlated with behavioral finding, and both are correlated with the state of the dendritic arbor and dendritic spine density. Okay, once what happens in hypoxia and how it is recovered in presence of the Bacopa extract. So this was done in CA3 region. Earlier it was in CA1 region, and this is in CA3 region. And we can summarize that neuron cell density in the hippocampus of hypoxic mice decreases significantly as compared to control and is recovered towards normal with pre or the post treatment of Bacopa extract. Dendritic arbor and spine density undergo significant decline in the hippocampus in the hypoxic condition, and administration of Bacopa monnieri extract reverses the effect of hypoxia on above towards normal. Okay. So in summary, we can say hypoxia lead to decline in the neuronal cell density and reduction in the dendritic arbor. Already I have mentioned this, again I am emphasizing, as well as spine density in which may be associated with decline in the memory. Bacopa extract treatment reverses above towards normal, which may be associated with recovery of memory impairment due to hypoxia. In conclusion, this is the one where I am coming to the end of the talk. Our study reveals the molecular mechanism of hypoxia-induced decline in memory and neuroprotective role of Bacopa monnieri extract involving synaptic plasticity associated proteins, metabotropic glutamate receptor 1/5 and EMPA receptor, which can help in designing a better therapeutic strategy to deal with the hypoxia-related mental health issues. Thank you for your patient hearing. If any question is there, I'll be happy to answer.
Good afternoon, sir. This is Arunima here from Chhattisgarh.
Yeah.
Sir, I wanted to ask one question. That in your presentation, you have mentioned that this Bacopa actually increases the density, CA1 density, as well as the dendritic density is also increased after Bacopa treatment in hypoxic mice. So, can it be presumed, or is there any evidence that it can be used for treatment of dementia or any other memory-related issues or conditions?
Yes, several memory disorders, either due to dementia, not only dementia. In our lab, we are working on several issues. That is a model of age-dependent decline in memory. The other reason is, Alzheimer's disease, which also leads to dementia, and loss of memory. So we are seeing similar kind of result in case of the dementia led by Alzheimer's disease. Also, I have not mentioned what kind of work and how much work is going on in our group. People are working on Parkinson disease at various levels, and they have found that Bacopa is helpful in restoring the neuronal cell density and also the dendritic spine density. So in case of human, Bacopa is being used for ancient period, and since mechanism was not well recorded, we have
Yes.
started working. The other people in country also they are working, and our finding on animal model, as much as possible, we can say yes. In case of dementia, it has been utilized by Ayurvedic practitioners, and uh, they have found positive result. But how much positive, for how long positive, these are all questions that one can only assume. But in animal model, we have found yes, it can be done.
Okay, sir. Thank you, sir.
Yeah, welcome.
And sir, one thing along with it, you mentioned that in case of Alzheimer's also, it has been found that it works very well. So in case of neuritic plaques, I mean, it
Yeah,
is possible to use Bacopa to remove them, or how it is neuroprotective, or how it is working against neurodegeneration. Can you just give me an information on what angles
I
should be?
can I can cite uh, just a part of our work which is going on, and partly it has been published also. I will not disclose everything,
No, no.
but but we have found that one of the components in the plaque,
Hmm.
that is beta amyloid 42. Okay, level of that decreases by Bacopa treatment.
Okay.
That means Bacopa is related with the processing of the precursor of beta amyloid peptide, that is called amyloid precursor protein. So APP processing is regulated by Bacopa. This is our primary finding, and a part of work that has been also published.
Okay. So I'll definitely go on the internet and check whatever publications are there for you.
Yeah, this is in an Indian publication. I don't know whether it will be seen there or not, but this has been recently. This was a primary data.
Okay.
This was published, and I don't know whether it is available on the internet or not. But this is on the this is on CMIG journal. Okay.
Okay, sir. Thank you.
Any other question?
Sir, one of our student has asked a question in the chat window. Sir,
I have
what
not
is?
seen. I
Sir,
have
I'm,
not seen
I'm
it.
I'm I'm repeating the question. Sir, what is the
Okay.
reason for post-treatment of Bacopa monnieri extract is not recovering significantly as compared to the pre-treatment? Is there
Yeah,
any antagonistic effect between them?
I don't know whether it is any antagonistic effect. I can only say that pre-treatment is better compared to the post-treatment. The reason may be the neurons, they are post-mitotic, and once their system is aggravated so much, and once the hypoxia has affected so much, then Bacopa may not be that much helpful. If the animal is pre-treated with Bacopa, that prevents the effect of hypoxia. So that could be one of the possibility, that hypoxia, once it has been there, post-treatment of Bacopa hardly helps the situation or improving memory, but pre-treatment prepares the brain to be less harmed compared to the hypoxic condition without Bacopa. Okay.
Sir, I have one query.
Yeah.
Sir, just I want to know, during prenatal condition,
Yeah.
does there exist hypoxia-like condition when the
Pre-
child is in the womb of a mother?
Yeah. Yeah. normally it all depends on whether mother is suffering from a condition that may lead to hypoxia. In a normal course, taking human as example, if the mother is well and if there is no other ailment in the mother, especially in the respiratory system, and also in the blood circulation in the placental system or placental barrier that separates the child from the mother, but that is not the physical separation, that is only because of protective separation is there. So if by any way mother's oxygen level is affected due to any reason, the child will be affected. The example I can give you, once the parturition takes place, okay, or during process of this, during labor process you can say, if there is so much of time of stay of the fetus in the uterine pathway or in the cervical pathway, service pathway, then the fetus is devoid of oxygen, and it has been reported that if there is birth delay, then the fetus can suffer from the lack of oxygen supply. And many of the children taking birth this way because of overstay in the cervix during course of labor process, okay, the children suffer from mental retardation. Some children, they have been described or reported to suffer from say speech loss. Some children, they have been described to suffer from the hearing loss. So varieties of problems have already been reported, and the major assumption has been during that process, what is the time gap? If it is too long, there will be too long stoppage or deficiency in the oxygen supply to the fetus, because then the placenta that was supplying oxygen, okay, that has been cut and the child survives on the aerial oxygen. Okay, so indirectly I want to say that if in the womb, if mother is affected somehow, say because of asthma or because of any lung infection or any other situation, when the blood is not properly flowing through the placental system, then the womb may be affected from hypoxic condition.
Sir, another question was with regard to neurodegeneration in hippocampus and recovery by Bacopa monnieri.
And
Sir,
Yeah.
sir.
So we cannot allow more questions. So this will be the last question.
Okay, sir. So is it the neurons are increasing or the hippocampal cells are regenerating?
I had mentioned during presentation that cells which have been dead, okay, they cannot come back. So the whole idea is that there is a recovery of the neuronal cell density. So this may be possible only because of the hippocampal neurons, they are maintained in the number by a process of neurogenesis that takes place in the dentate gyrus. Okay. It is not because of regeneration. Regeneration means the neuron has been degenerated and then the process of regeneration has taken place. So the proper term is not regeneration. The term here should be neurogenesis. And neurogenesis means the neuronal stem cell, that is resident neuronal stem cells in the hippocampus, that may come up to be differentiated into neurons.
Thank you, sir.
So thank you very much, Prasad, for
So is there any question?
informative lecture.
Further question?
सुबोधन सर आपकी आवाज
Rajkumar, you are telling something.
Oh, no, sir. Subodhan sir is
Yeah,
saying
Professor Jain, yeah.
Okay. So for this session is going to be closed now, and before closing it, I request The report here, Dr. Rashmi.
I am unable to hear your voice, Professor Jain.
I don't know why this one is coming, but I used this one.
I am not hearing you.
Okay.
Yeah, now it is.
Okay, on behalf of the organizing committee, being chairman, I am thankful to Professor Sukala Prasad. And now I request Dr. Rashmi Srivastav to sum up this session, please.
Before I would like to thank the organizing team, especially Professor Ahi, Dr. Raj Kumar, and Dr. Rasmi as well, and Professor Jain for giving me this opportunity to present a piece of work on a particular area of hypoxia only. Thank you so much for giving me this opportunity. So, Dr. Rasmi.
We also thank you, sir.
Thank you, sir. Thank you, sir. So in this session, we had two talks. The first one was by Professor Jelena Radulovic, and talk of Professor Jelena was really very interesting. She explained how psychological processes are intimately linked to the brain, which is of course a very strong electrical network of neurons having synaptic connections. She told about the molecular network with microRNAs, how the single molecules can perform complex computations, and how these changes in all this network can affect the behavior. Now, the psychological processes are about dealing with all these informations. So she very well explained about the genetic models and approaches for animal models of psychiatric disorders like schizophrenia and so on. She highlighted about the animal models of studying depression, the knockout mice, are related to this behavior, the oxytocin knockouts, which are very relevant for autism, then schizophrenia, which is a disorder of reality perception. So she discussed so many things. We had a very good useful information about the molecular mechanism of behavior and psychological processes in her lecture. The second lecture was
Not audible.
Cognitive impairment and neuroprotective effect. And he explained us how the cobalt chloride acts as hypoxia mimetic agent, and how this hypoxia condition can be maintained, about HIF1 alpha and beta, how they bind and form complex in normaxia and hypoxia. And he told us about several ways to develop hypoxia models, and how the injury to brain due to lack of oxygen can lead to impairment of learning. He highlighted various neuronal system crosstalks for achieving the cognitive function of brain, how glutaminergic system is affected by hypoxia, and AMPR has the potential of in learning brain functions. So he told about the function of Brahmi, which can boost the memory enhancing. Brahmi can play a neuroprotective role in hypoxia conditions. And further, he also showed about us the decreased expression of glutamine receptors in hippocampus in CA3 and CA1 regions, how they're responsible for spatial memory and due to hypoxia conditions. And he clearly showed us by experimental method the molecular findings related to behavioral findings. We are very thankful to you, sir. Thank you so much for your talk.
Yeah.
Thank you, sir.
Thank you so much. Thank you so much, sir.
Thank you.
Thank you, Rashmi
Thank you
ma'am.
very much.
Okay.
Okay, I'm going to Banaras.
We will meet after lunch break at 1:30 for session 2, oral presentations.
And we will be meeting for the next session by 1:30. And now this session is over. Thank you very much.
participants who go to will meet again. We'll resume the session from 1:30 for oral presentation.
Sir, बोल दिया मैंने.
नहीं और बोल दो एक बार और बोल दो.
We will start again at 1:30 for oral presentations.